Okay. Okay, great. Welcome back, everybody, to the Marine Mammal Commission 2023 annual meeting. We have on the agenda today, first, a recap of yesterday, which is going to be very brief. First of all, to tell you that the proceedings and talks of the meeting are being recorded, and they will be up on our website fairly soon, so you don't have to feel like you missed anything. We get the talks up as well, the PowerPoints as well. The structure of the meeting, as we talked about yesterday, has been going through four elements of marine mammal response and effects of climate change on marine mammals. Yesterday, we talked about ecology and ecosystem dynamics, started with a session on shifting distributions, prey, and threats, and then an afternoon session titled basically the same topic, but habitat alterations. And today we're moving into marine mammal health and then into a study or discussion of the implications of this for stock assessments of marine mammals. So that's a very brief recap. I thought about going through every talk and spending about 4 hours on it, but I decided not to. So the only thing I wanted to mention, those of you who got your Washington Post on your front step this morning, saw that the Biden-Harris administration has released the 5th National Climate Assessment and has announced more than $6 billion to strengthen climate resilience across the country. I won't go into great detail on this. I want you to look it up. I think we started our meeting talking about how we were following the president's climate agenda in developing our papers and discussion on marine mammals and climate change. So basically this meeting is part of the administration's efforts on climate change, and we really appreciate your contributions to that. We've really benefited from the discussions and know we'll benefit from the discussions today. And it's really helpful to frame this. We have lots of issues with marine mammals. We have entanglement, we have vessel strike, we have harmful algal blooms, we have all we're hearing about today, but to frame it in the frame of climate change, I think really gives us a new way to talk about it and sort of cuts across some of the divisiveness of the issue. So let's get going. I want to introduce Randy Wells, who he pointed out I met like 47 years ago, and we have, was it 47? I can't maybe 90. Anyway, Randy, take it away.
Good morning. It is truly a timely session that we're going to be having today. Over those many decades that Peter and I have known each other, things have changed a lot in terms of how we look at and try to conserve cetaceans and other marine mammals, going from focusing on mortalities primarily to now trying to look at other issues relative to these animals, sublethal effects, and especially the recent, in the last couple of decades, focus on looking at the health of these animals and how that can be used to try to provide conservation measures for them. We're fortunate that in the room today, we have many of the pioneers of marine mammal health studies, health assessments, and we'll be hearing from some of them in the panel and hopefully hearing questions from others as we go through the morning. Our first speaker today will be introducing One Health and potential climate effects on population health, and it's Francis Gulland, our esteemed commissioner and a research associate at the University of California, Davis.
Thanks, Randy, and thank you all for being here. And Brady, can you have the first slide, please? Thanks. So I'm really just going to introduce this session, and we're going to hear more about specifics from the subsequent speakers. And the title is One Health, and then Marine Mammal Health and Climate Change. And before 2019, I should say that One Health was really this sort of obscure concept that really veterinarians referred to when we were trying to get funding from the human medical community. But since 2019, with the emergence of COVID, I think we're all very aware of how coronaviruses can infect animals and then jump to humans. And this transmission between animals and humans is influenced by the environment. So now One Health has become very much a parlance across government agencies. It's not limited to the world of NIH, CDC, and vets trying to get funding, but I think everyone talks about this And really, it's the concept that both animal health and human health are intimately linked and that they're both influenced by changes in the environment. So fine to say, but what does it really mean practically other than the coronavirus pandemic? And in the mammal and sort of ocean world, I've worked on domoic acid for a while, actually for 20 years now in California sea lions. And there the sort of idea was this was a classic example because the environment changes, we get increasing harmful algal blooms. They can accumulate in seafood that humans eat. They can accumulate in seafood that mammals eat. So what signs and pathogenesis and changes in the brain we see in sea lions could also occur in humans if they ate similar doses. And these are influenced by the environment. So that was a nice sort of, oh, we work on One Health. This is what we study. But actually, we heard from Karen Forney yesterday that it's far more complicated than that. It's not that there's just one little tweak, one little change in a harmful algal bloom, and then we see a predictable change in either human health or in sea lion health. And so for those of you who weren't here yesterday, this is a slide summarizing changes that happened in California, resulting in increased entanglements of humpback whales, but triggered by climate change that influenced changes in the prey distribution of the humpbacks, increases in harmful algal blooms that resulted in a delay in the crab fishery being opened in California, which then resulted in an overlay of the crab fishery and humpback whales along the coastal California. So very much not that simple Venn diagram of three circles and one little nice thing in the middle. It's a much more complicated picture. So that's simply domoic acid. So I think that the reality is that when we think about climate change in the One Health sort of paradigm, we know that we have a whole series of cascading effects of climate change. Climate change is going to influence physical changes in the ocean, and these in turn will have sort of abiotic responses. And then these will be manifested in changes in orange that we heard about yesterday. So today we're going to move on to the black boxes and how this series of changes resulting from climate change can result in changes in individual animal health, but at the population level, changes in reproduction and mortality, either due to the changes in health or directly from changes in prey distribution, result from these changes in climate change. So it's really a lot more complicated than the simple One Health picture. But for those of us who are trying to work in the funding realm, the One Health idea has really given us opportunities to work on understanding the impacts of climate change if we set that within the One Health framework. And I think we'll hear a little bit more of that later from Jonathan. So yesterday we heard about really effects via prey. Today we're going to focus on the health, the middle of these three black boxes, and how can climate change really change health of individual animals. And I think the two main routes, if we don't think about prey and straightforward nutrition, we could put that in a sort of separate bin. When we think about health, it's really by being impacted by an infectious disease, a pandemic. And in the mammal world, our coronavirus are really the mobiliviruses that cause just dramatic epizootics. And currently, avian influenza is killing tens of thousands of marine mammals in South America, mostly otariids, and currently today, elephant seals in Argentina. And then the other way that climate change is impacting individual health are through increases in exposure to toxins from harmful algal blooms. So we have these two mechanisms that health is impacted. And when we think about the pathogens themselves, these are viruses, bacteria, fungi, and the protozoa. Climate change can either increase the survival of these pathogens that are sensitive to temperature. We have a few examples of those. Or it can really just change the distribution of either the vectors or the animals themselves so that they're more likely to get exposed to pathogens that have been around, but are now have possibility to get into marine mammals. So the first, increasing harmful algal blooms, we have more evidence of these impacting marine mammals. So back in 1998, domoic acid toxicosis was first identified as killing California sea lions. And that was a really dramatic event. It was Memorial Day weekend in May 1998. It was actually it was a ocean, something like an ocean year of the ocean, and within that it was the weekend of the ocean, and Bill Clinton was in Monterey and CNN was filming him, and as he walked out across the pier at California Sea Line, had a seizure, aborted a fetus, and it popped into the ocean right there on CNN. So that led to a huge amount of media attention of, oceans not so blue, red tides, and I always have my little joke about, I wonder if Clinton ate some anchovies and then couldn't remember. But anyway, so since 1998, when that was first occurred, we've seen increasing impacts on California sea lions beyond just seizures and death, but effects on reproduction, effects on long-term memory, spatial ability. And then along the California coast, we've seen more impacts on other species, northern fur seals, Guadalupe fur seals, sea otters, and now cetaceans. And this year there was a huge bloom in Southern California. In the rest of the world, domoic acid producing blooms are also increasing, but it's been harder to link the effects of the blooms to actual mortality of marine mammals. And that's really because of the limited ability in some places where these blooms are occurring to do marine mammal health surveillance. So you may have seen a couple of years ago, there was a large number of dead fur seals and sea lions off in Namibia and South Africa. And at the time, there was a massive domoic acid producing algal bloom, and domoic acid was reported in water samples and in prey. But unfortunately, there weren't necropsies of those animals to really explore how much of that mortality was due to changes in prey distribution and how much was due to domoic acid. Similarly, in Europe, there are domoic acid producing blooms around the UK, and Elsa Hall has been detecting domoic acid in harbour seals around Britain. So domoic acid producing blooms are they're becoming more common, more widespread, longer lasting, and also more toxic. Another bloom that we know impacts marine mammals are saxitoxin producing blooms. And back in 1987, Joe Girassi was the first to recognize that humpback whales were dying off Cape Cod with high levels of saxitoxin in tissues, and there was high levels of saxitoxin in mackerel. So this was right in Cape Cod, where there are high numbers of, right now, we worry about right whales feeding off Cape Cod. That was 87. Since then, the largest die-offs of whales anywhere in the world have all been linked to saxitoxin. So off Chile in 2015, there were over 300 dead sei whales detected. Again, logistics were challenging because it was a remote area, so very few animals were sampled, but those that were had high levels of saxitoxin in the gastrointestinal contents. So that bloom was probably associated with the increase in aquaculture around southern Chile, fostering right nutrient composition for saxitoxin producing blooms. And then last year, off Punta Valdez again, there were large numbers of dead adult female southern right whales with high levels of saxitoxin in. So we're getting basically more evidence of these blooms killing marine mammals, and these blooms are increasing globally. Then moving on to the pathogens, this is a little bit more challenging, and we have really limited information so far on really the impacts of climate change on pathogen exposure in marine mammals. There's a lot more information from the terrestrial wildlife field. So we're sort of limited to, when we think about, we heard about polar bears yesterday and changes in their distribution. And we also heard concerns about ice seals perhaps using land more for birthing. So if a marine mammal comes ashore, it's going to be exposed to terrestrial pathogens. So there is some work describing an increase in antibodies to terrestrial pathogens in polar bears, which suggests if they have the antibodies, they must have been exposed to the bug. And then other work from Alaska has found that Vibrio parahemolyticus, which is a bacteria that is not only temperature sensitive as far as its survival, but it needs temperatures of over 15 degrees centigrade to actually produce toxin. So this is essentially cholera poisoning. This was first detected in people in Prince William Sound. And then that increased the sampling of marine mammals. So they directed their sampling to look for Vibrio, having heard that it was in humans. And Kerry Gertz and her group found this Vibrio in fecal samples from belugas, otters, and hubberpoppers in Alaska, so thus expanding our known understanding of the distribution of this organism. What it does to those animals, we still don't know because this was a survey of fecal samples, but it does cause, it causes cholera, it causes diarrhea, and it can be fatal. There are also new pathogens that are emerging associated with changes in climate. So there's a fungus called Cryptococcus gattai. This is a fungus that survives in damp, warm conditions and has been, its distribution around the world has been linked to the distribution of eucalyptus trees. And really surprisingly, the greatest number of cases per capita in humans are in the Pacific Northwest and specifically around Vancouver Island. So historically, it was a tropical disease following the distribution of these trees. And now with warmer winter temperatures, which allows survival, overwinter survival, and thus increase in numbers of this fungus, not only have there been cases in humans of Vancouver, but Steve Rafferty and his team have identified cases in harbor porpoises and Dall's porpoises, and it causes pneumonia, abortion, and encephalitis, and generalized infection in marine mammals. And there have been over 300 cases just in the Pacific Northwest. So this is a pathogen that we had not seen in the States at all in marine mammals. And then this increase or sudden emergence of this disease was associated with these warmer overwintering temperatures. So that's really the limited amount of actual case studies that we have. The reason we have such limited information is that we haven't done a good job so far of linking health changes with environmental measurements. And I'm looking at Sue because it's something we talk about a lot. We have the world of measuring the environment, and then we have the world of vets trying to treat or cut up dead animals, and the two rarely come together. And then secondly, we have very few large-scale systematic sampling studies for specific pathogens over time. So in the example of the Vibrio, because there was a human case, the group at the Sea Life Center, next time they were handling live animals for other reasons, they collected some fecal samples and then said, let's look for Vibrio. So we don't know if it had been there for 10 years, 20 years. We don't know if it's increasing. We don't know if the distribution is changing, because to find it, you have to set up specific culture conditions. So we really need to think about this large-scale sampling where we look at the distribution over time. It's a bit like stock assessment for bugs. Whole new realm, but to put it into that parlance rather than individual case reports. So because of this lack of sort of baseline information, it makes predictions and also response preparedness difficult. We're still very much in this reactive mode is, yikes, we've got all these seizuring sea lions, we've got dead dolphins, what do we do? And we go into sort of panic mode that we shouldn't really need to have to do. So how do we move forward from here? Well, I think we're going to hear some examples from the other speakers, or I know we are. We will hear some great work. But we really need to think about population level health surveillance as distinct from single case investigations. So that means not being the single vet that responds to an animal on the beach, but how do we integrate the veterinary community with the larger scale marine mammal world of stock assessments that we'll hear about later, and integrate health into numbers of animals and numbers of deaths and numbers of animals not reproducing. And then we also need to think about some targeted studies of climate sensitive pathogens. There are organisms that we know are going to be changing or are changing with climate change. And for that, I think we need to really work closer with our terrestrial wildlife disease colleagues, because they have already larger data sets and I would say a more advanced understanding of some of the organisms that are changing. So, for example, West Nile virus is a virus transmitted by mosquitoes. Jonathan's nodding. We have very good understanding, or better understanding, of how that virus is spreading with an increase in the range of the mosquitoes. And we do know from a few examples of cetaceans that have died in zoos as the virus moved across the US, that marine mammals are very susceptible to it. And then finally, I think we really need to start focusing on how to integrate environmental data, animal distribution data, and then health and causes of death, so we can track these changes. we've talked about it for a while. We have a lot of advancement in the IUS world. We heard from Clarissa Anderson yesterday about her work to predict algal blooms, but that's still in a separate arena from the marine mammal health sort of investigations. So I think several of us have been thinking about this path forward. How do we get a better understanding of the impacts of climate change on marine mammal health specifically, so that we can be better prepared? So this is a slide from Sue Moore, but we really need to start thinking about how do we look at these 3 steps? How do we integrate marine mammal health monitoring and surveillance with an understanding of our and our monitoring and surveillance of ecosystem processes? So with that, I'm going to stop, and I think we will hear from other speakers on these specific examples, and then we can have a discussion with a panel about how we move forward. Thanks, Randy.
Thank you, Francis, for a great introduction. And I'm not getting control here. Where is it? Okay. Our next speaker this morning will talk to us about Hawaiian monk seal research program. It is an exemplar of a successful conservation medicine approach, and it's Michelle Barbieri, the supervisory veterinary medical officer of the NMFS Pacific Islands Fisheries Science Center. Okay, Michelle, now that I'm mastering the technology up here.
Good morning, everyone. It's really an honor to join all of you here today. I realize this is an extraordinarily lengthy list of co-authors, but when the suggested title for your talk speaks to being exemplars of successful things, I think it's really important to acknowledge that that success is built on the shoulders of so many. So I've listed many of our Hawaiian monk seal research program members, past, present, and honorary. And there are many more seasonal field researchers and partners that I didn't have room for today, but really want to acknowledge those contributions. So a quick review of the Hawaiian monk seal from what we heard from Jason yesterday. They are endangered. They're one of only 2 mammals endemic to Hawaii. They're the only coral reef associated seal and the only living species of their genus, Neomonachus. They live here in the areas circled in blue in the main and northwestern Hawaiian Islands, and their range also includes that little blue dot at Johnston Atoll. And in the most recent estimate of the population size in 2022 was just over 1,600 animals, and they have been increasing at an average of 2% per year since 2013. So today I'm going to tell you about how we apply conservation medicine in this species, and I thought I'd introduce that by breaking it down into 3 main areas: observational data, population health surveillance, although I wish I had a chance to edit my slides because I think stock assessment for bugs, Francis, was a great way to talk about that, and also interventions. And so each of these informs another. And if there's one thing I hope you'll take away from my presentation today, it's that baseline information, and in this case, specifically that on health, has really allowed us to open the door for Hawaiian monk seals to some bold pursuits and becoming the first in the world to vaccinating a wild marine mammal at the population level. So what am I talking about with these 3 pillars, and how have they helped us do that? So first, there's observational data. This can come from skilled field researchers, from partners in the Stranding Network, from conservation groups, from communities. The public is a great resource of citizen scientists. And these data are really powerful. So they tell us about things like body condition, which we heard a bit about yesterday, behavior. They can also tell us about wounds or changes in patterns for individual seals. So we can think about the emaciated seal on the right and the apparently healthy weaned pup on the left. And at that individual level, we compare observational data that we get on those things with a seal's individual reproductive history, its stranding history, and we can look more broadly at environmental data like local conditions, or even in some cases, larger oceanographic variables to provide context. And then we can zoom out to look across those observational data at the island level, the island group level, or even the archipelago level. And collectively, that can help us inform our understanding of the scope of a given issue. And observational data gives us important clues to threats. their impacts on survival of animals, and enable us to monitor outcomes and pick up on trends as they emerge. Most importantly, we use these data to design and measure the effectiveness of life-saving interventions. For example, we can reunite mother seal pups using our observational data. And then we can do things like stand up rehabilitation efforts and conduct survival enhancing translocations if we see areas where seals are skinny compared to areas where seals are doing okay nutritionally. Right. Sorry, I think I failed to advance that slide. Sorry about that. Moving on to stock assessment for bugs. In this example, this is directed research on seal health. And for us, a lot of this is typically hands-on. Designing standard sample collection protocols is really important to do across all the animals we have the opportunity to handle. And I want to emphasize that because it forms the foundation of our understanding of disease threats in this species. So we work with partners. We ensure that we're collecting standard samples, whether it's a seal that needs rescue or a healthy seal that maybe we're handling for other scientific purposes like instrumentation or tagging. And additionally, we have collection protocols for postmortem examinations and do our very best to sample every animal that we possibly can. So together, those routinely collected samples are analyzed for pathogens that we know exist and occasionally some things we want to make sure we're monitoring for, just in case they show up. We archive samples for longer term studies and to enable screening outside of those standard protocols and to provide material for future questions that maybe we aren't thinking about right now, but we can go back and pull those samples later for something that we might need. And a timely example of this, I think, is avian influenza. So that's something that Hawaii has not seen, but is of worldwide interest right now. We do have at least that backbone of surveillance capacity sitting in our hands. And so we have an archive of serum, we have prospective sampling regimes that we can modify as things go forward and as we better understand that risk. So for both of the seals in these pictures, one in the wild and one that will probably be more readily sampled in rehabilitation, we're screening and checking for the things that we need to be aware of, partly to inform the care of sick seals, but also to give us a foundation of health data that lasts across the population. Really, we can't know what's abnormal until we know what's normal. So together, these two data streams inform many of the interventions for monk seals that we design and conduct and evaluate for effectiveness. And at that individual level, those interventions include monitoring a seal, maybe to determine if intervention is needed. Perhaps in some cases, benign neglect is the most appropriate thing. Or we may choose to intervene to mitigate a threat. And this can be, again, rehabilitation or something more advanced like the translocations I mentioned before. And as I'll emphasize again later, the power of observational data can also be that early warning system so that if there are new threats or new diseases that emerge, we can take action to minimize their impact or their spread to adjacent seals. And we can use hands-on opportunities to collect samples for further health surveillance on live and dead animals according to those standard protocols. One of the things here that comes to mind is also maybe not just in a specific pathogen context, but for contaminants. We don't measure contaminants in monk seals routinely, but we're always trying to make sure we're banking samples to complement the studies that have been done in the past so that we can look to see if there are changes in the future that might impact health. And we've designed quite a few interventions in response to what we've learned from these data streams. I've mentioned many already, but we can also be thinking preventatively. And that's where these data have really helped us take some solid steps towards disease prevention. So one of the concerns that we've already heard about briefly today so far is the threat of morbillivirus outbreaks. This virus is similar to distemper in dogs, measles in people, and it's primarily spread through respiratory secretions. And it's caused outbreaks on the order of thousands to tens of thousands of pinnipeds and other species in other parts of the world. So our long-term population health surveillance efforts were really important here in terms of developing a strategy. So first, we had that baseline data that indicated that there was no evidence of prior exposure to morbillivirus. And so the species didn't really have any pre-existing immunity that we could detect. And you now already know that it's a small population. So to put it in context, the total number of monk seals, Hawaiian monk seals on the planet, is smaller than the number of animals that have died in some of those morbillivirus outbreaks in pinnipeds and other places. Monk seals also have poor genetic diversity. They occur in a region where morbilliviruses have been detected in stranded cetaceans, and they're somewhat regularly exposed to unvaccinated dogs on public beaches in certain portions of their range. And in other parts of their range, they are very far away from observational view, at least for about 10 months out of every year when researchers are not there to monitor them. So in times when climate change is impacting the movement of animals and subsequently pathogens like morbillivirus around the world, disease introductions and outbreaks are a growing conservation concern. So we've developed and implemented this vaccination program for morbillivirus, but a lot of work was really needed to take this from concept to reality. We looked closely at monk seals from an epidemiological perspective, and this is another juncture where our long-term monitoring program provided key insights. So unlike a lot of pinnipeds, monk seals do not haul out in dense aggregations, but they do interact regularly. So we first wondered if there was sufficient contact for an outbreak. And Jason Baker led the development of a morbillivirus outbreak model using a susceptible, exposed, infectious, recovered model. And in that, we used things like the literature to make assumptions about variables like the latency period and the infectious period. And we used our decades-long experience to make assumptions about things like how seals would move between different compartments of that model and how long, for example, until we could practically detect the first infection. So we also used observational data that we had on the species to develop a social network model, and that enabled us to account for differences in contact rates that we knew existed between different age and sex classes of monk seals. And we ran that to simulate an outbreak, and then again with the addition of vaccination by various strategies. And as Karen pointed out yesterday, and so eloquently reminded us, a model is just a model. But it did help us conclude that, yes, there is at least sufficient seal-to-seal contact within this species for an outbreak to occur. And we could infer that a strategy where we vaccinated seals in response to the disease being introduced to the species was not going to be sufficient to stop its spread and outbreak proportions. But we could reach herd immunity if we vaccinated seals ahead of that outbreak. So the chosen mitigation strategy is to prophylactically vaccinate seals, combined with preparedness efforts to detect and remove infectious seals from the population as early as possible. We tested and now use a commercially available distemper vaccine made for ferrets, and seals get 2 shots about 4 weeks apart, as was tested in seals in human care prior to use in the wild population. And since 2016, over 900 seals have been vaccinated, over 1,800 injections have been given, and we have not had any adverse reactions. So going forward, NOAA and its partners want to foster community awareness so that sick seals can be detected early, which will provide opportunities for early intervention. We know we're probably not going to identify patient 0, but our goal is to get as close to that as possible, at least in areas and seasons where we have seals and people overlapping. In addition, we continue efforts in population health surveillance to maintain a baseline finger on the pulse of the species population health. And in the future, I really hope to have point of care diagnostic testing available for our remote locations. And hopefully at some point, this work may prove to be useful and have some transferable application, whether it's to new disease threats for monk seals or to other taxa. And as I wrap up, I'd be remiss if I didn't mention that this vaccination effort is one of many targeted interventions listed here that are conducted as part of our recovery efforts for this endangered species. It takes an ohana of partnerships to make this work possible, including our agency partners like the state of Hawaii, the US Fish and Wildlife Service, and non-governmental partners, especially the Marine Mammal Center and the Papahanau Mokuakea Marine Debris Project. So Jason mentioned this yesterday, but I think it's really important to call attention to the Silver BB study that Bert Harding and others have led. And that work has shown that these interventions have improved the survival of seals such that 1/3 of the population alive benefited from a life-saving intervention or is the offspring of a seal who did, meaning that these interventions have a multiplier effect into the future. And we don't really know the payoff of the vaccination program yet. I think a lot of us hope we probably never really will. But I do think it's important to point out that with proactive approaches, that does, and a foundation of baseline population health information, we can use that to look to the future. We're going to have a lot more complex issues to tackle, like the habitat loss that you heard from Jason yesterday. We're also dealing with terrestrial pathogens like Toxoplasma gondii, which affect native wildlife, humans, and really are a One Health issue. So through my talk today, I hope I've been able to give you a little glimpse into one of the many things we're doing to safeguard endangered Hawaiian monk seals through the lens of our conservation medicine science. and that we can serve as an example of a bold idea being turned into a proactive approach with careful planning and a bit of courage. So that gives me a lot of hope that as we look towards climate change and issues that are going to tackle even stronger levels of collaboration and cooperation and outside the box thinking, that there are some opportunities for us to move forward and protect these animals. So thank you very much.
Thank you very much, Michelle. It's an amazing effort, and it's so encouraging to hear that there can be successes in this way. And again, I'm not getting any response to the touchpad here. Yes. Okay, Chris, can you hear me?
Yes, I can hear you. Can you hear me okay?
We can. All
Perfect.
right. And you'll put her on from up there?
How's that looking to you all for presenter view?
Okay, so we're going to be hearing from Chris Cruder-Johnson, from the professor of medicine and epidemiology from the University of California, Davis. And she'll be talking about southern sea otter recovery, exemplar of climate-related contingencies. There we go.
Thank you so much, Randy. It's a privilege to be here, and I've really enjoyed the presentation so far. So as Randy said, I'm going to be talking about southern sea otters and recovery. And southern sea otters are, of course, a very functional part of the ecosystem. They have keystone activities that sort of dictate the nearshore system. Certainly along the central coast of California, this has been well documented, and numerous studies extending up along British Columbia and Alaska that also show that where sea otters are present, there is a healthy system that includes kelp, which is, of course, critical habitat for so many fish, invertebrates, and other marine mammals and birds. So, I think in terms of the importance of the kelp ecosystem, we're learning that that is very, very, very essential for sequestration of carbon dioxide and contributing to climate regulation, and one of the ecosystem services that the ocean provides for climate regulation, and also an important functional part of the ecosystem when we think about eelgrasses as well, in terms of controlling nutrient pollution. And sea otters, basically as this top predator, will take care of overgrowth of algae that tend to smother eelgrass by allowing these sea slugs to exist in heavily polluted systems such as Elkhorn Slough, which is off the central coast of California, which is a very nutrient-rich environment that tends to have a lot of algae. And so sea otters, by predating on crabs, are able to allow sea slugs to exist in that system to help control the algae overgrowth. And then, of course, very well known from work all the way from Alaska down to California, that sea otters predate sea urchins, and when they do that, they allow a balanced ecosystem to prevent overgrazing of kelp by sea urchins. So, like many marine mammals, sea otters are a really excellent indicator and a direct reflection of their environment. And sea otters are perhaps a little unique in that they very, very closely can reflect some seriously localized processes in the nearshore system. That's because they're such a highly visible marine mammal. I think those of us who've been fortunate to work with sea otters feel extremely lucky because they have such a near shore distribution. They bring their prey up to the surface, which is really convenient for us to be able to see what they're eating. And also the females are fairly site fidelic, so they'll exist in the same location with a relatively small range for much of their lives. And so this has enabled really intensive study at the individual level that has helped foster these huge collaborations across agencies and academics that we're able to look at individual animal movements using telemetry and understand individual foraging behaviors and specializations on types of diets, their daily movements, their use of the range, and look at their range use across different age groups, as well as, of course, understand reproduction and survival at the individual level. And combined with that, As Francis was saying, is true for many marine mammals, also true for sea otters. There's a fairly robust carcass recovery program with veterinary pathologists who will look up as many animals as they can to be able to classify cause of death for those. And that's been in existence with USGS and the California Department of Fish and Game for over 20 years. And so the population health of sea otters has been able to, for us, in California, certainly directly reflect these spatial and temporal changes that are in the nearshore system and how that affects certain aspects of disease and other conditions that we've seen in sea otters. And so sea otters have had protected status for over a century. They were very, very closely hunted down to near extinction because of the fur trade, very uniquely prized fur because of the thickness and that really resulted in estimates are over 800,000 sea otters that were harvested in the 1700s up through the 1860s until basically there were not enough otters to be able to harvest anymore. That really closed down that aspect of the fur trade, and they were easy picking because they're so close to shore and because they do float at the surface. And essentially by 1900, there were only small remnant populations. The northern sea otter population, subspecies across Russia and Aleutian Islands, and then of course in Central California, they were thought to be extinct, and then 50 individuals were detected just off of Big Sur, that then became what we know as the southern sea otter subspecies population. These are the international and federal protections that came in. So the 1911 International Fur Seal Treaty, of course, protected them at the time when there was very few, but they still needed protection. And then in the 1970s, the Marine Mammal Protection Act, and in 77, the Endangered Species Act protected sea otters when the population started to rebound and was really, really worth protecting at that point. Southern sea otters have expanded their range because of these regulations, and they have done so, and I'll show a figure in a moment, really impressively with very, very important linear growth across much of the past century. And there's also been direct efforts and interventions similar to what Michelle was talking about, where there's been a surrogate pup rearing and release program that was established by the Monterey Bay that's been highly successful at releasing individual pups that would not have survived on their own. And there's been also really important reintroduction efforts for populations that a little bit in California, where we've tried to move populations out to San Nicolas Island, for example, and other relocations to get them out of harm's way where there's fisheries that are active. But importantly, for the northern sea otters in Washington, British Columbia, and Alaska, it's estimated that those reintroductions actually have, at this point, accounted for 35% of the sea otter population globally to help that recover. So here's sort of the population trends recently for southern sea otters in Central California mainland, as well as San Nicolas Island, with that translocation effort just being shown up here. And you can see the really impressive a success story indeed for sea otter recovery with running averages. And then we just have not been able to do the census in the last couple of years because of COVID and restrictions on plane availability, and then sort of a reevaluation of how to do the census based on how best to count the numbers of otters now that they're around the 3,000 range, where we need some really specific estimates. So where are we now? Sea otters are about at 3,000. It wavers just above and then just below in recent years based on counts that have been able to happen. They seem to be at or near carrying capacity in Central California and are most likely food limited with the way that they specialize on prey and utilize the nearshore system. There's been limited expansion to the north and south over the last 20 years. And most recently, there was a 5-year review that Fish and Wildlife Service did, and they were able to retain their protected status under the Endangered Species Act for several reasons that I just wanted to talk about here. And one of them is because they really just occupy this central portion of the California range. Again, they've not been able to distribute further north or south, and this is a very small fraction of their historic range. Any large oil spill could take out a bunch of the population quite easily, so that remains a pressing threat given the shipping lanes for oil just off the coast. There's also high levels of shark bite mortality that have prevented this range expansion. And so sea otters being attacked by sharks has been a very important cause of mortality always, but has been increasing. And I'll talk about that in a minute, but really that has thought to be limiting expansion. So sea otters that make attempts to move up here get taken out by sharks here, and similarly just down south. There's also, as Francis mentioned, numerous causes of mortality, including biotoxins, and there's still mortality in fishing gear, as well as a lot of recreation off the Central California coast that inhibits some of their recovery. And of limited genetic diversity, as I said, they started from that very small stock initially, and we're starting to anticipate major new risks due to climate change. And so I wanted to talk about just a few examples of what we're expecting and what we've started to observe already due to climate change. So one of the important things that we've been able to document with sea otters and their nearshore distribution is the importance of these protozoal pathogens, including Sarcoidosis neurona and Toxoplasma gondii, that uniquely cause protozoal encephalitis or neurologic disease, brain-related disease, and symptoms in sea otters. And both of these pathogens have been introduced. They're non-native hosts or reservoirs for these species, cats and opossums, for Toxoplasma and Sarcoidosis respectively, that are not supposed to be along the California coast, and sea otters and other marine mammals are uniquely susceptible, sort of immunologically naive, and it is an important cause of death. It's been linked very closely to runoff and big storms that facilitate sort of this unfiltered runoff, especially in locations like I was talking about where eelgrass and other nearshore systems have been disrupted by human alteration of the habitat. And so we have seen clusters of mortality, especially due to Sarcoidosis neurona after big storms routinely in the springtime, and anticipate as those storms and that runoff is an increasing trend that we would see more of this in sea otters over time. We also, of course, have the issue of harmful algal blooms and just an absolutely amazing amount of work done by a huge number of colleagues across the California system in many species using that multi-species approach that was really important to be able to document, spearheaded by the Marine Mammal Center, documenting sea lions as one of the first indicators for harmful algal blooms, especially domoic acid, that for sea otters, I think really tends to have a unique manifestation of several different types of issues, though they have likely longer term exposure than many species, because domoic acid can persist in some of the shellfish prey that sea otters uniquely like to prey upon. And so when they have acute large doses, they do manifest similar to other marine mammals, where they'll have this sort of fatal neurologic disease, seizures and such. But they also, and we're increasingly document, have these subchronic and long-term conditions that end up causing death over time, but very hard to directly articulate to domoic acid. But through epidemiologic work that we've been doing, increasingly showing that heart disease, which is a really important cause of death in sea otters, is quite likely linked to chronic and repeated exposure to domoic acid in the nearshore system, with most otters exposed to domoic acid at one point or another. And so, of course, as Francis was saying, we expect harmful algal blooms to increase in both magnitude and persistence, as well as frequency, with warming trends in the oceans. And then for shark attacks, the evidence for a link to climate and the unfortunate consequence of warming trends is certainly a case study that has been revealed over the last 15, 20 years or so. We know that shark bite became the #1 cause of death in sea otters starting in 1998 over the next 10 years. And as I said, it really limited expansion to the north and south, and then work into and collaborations with folks who study shark distributions found that the sharks were utilizing the nearshore waters. They were following prey and pinniped availability, quite likely, but also changing their abundance and their distribution related to climate and sea surface temperature. So from 1982 to 2013, the upper range limit was Santa Barbara. And then they've increasingly moved into the nearshore system. And certainly juvenile white sharks have been found within Monterey Bay. And that was linked to the marine heat wave that occurred in 2014 to 2016. And so future climate conditions we expect will enable these juvenile white sharks to be closer to shore and to be in habitat that sea otters require. And then also for climate effects that are going to affect kelp cover, which is essential for sea otters to be able to protect themselves in that canopy, to be able to prevent shark attack. Those conditions could also make sea otters much more vulnerable if the kelp cover was in any way affected by expected changes in climate in the coast of California. And as Francis very eloquently said, there's also things that we're learning on the human front that we expect to be an issue for marine mammals. There's certainly warming trends and links to Vibrios, several different Vibrios that are pretty pathogenic bacteria that exist in the ocean. It's been sort of a opportunist infection in many species, including sea otters. But when sea otters are attacked by shark, for example, that is one of the bacteria that gets in. And if the bite didn't kill the otter, this bacteria, one of these Vibrio vulnificus or Vibrio parahemolyticus, and a new species that we found, Diabolicus, in marine species, would most likely be an important cause of death in those individual animals. And so we have documented these species in sea otters and antimicrobial resistance, unfortunately, in sea otters, in their prey, and the environment through sampling that we've done here in Central California. And so we expect these warming trends and what we see off the Central Coast to start to manifest up north. And there's certainly increasing cases in humans in areas of the US and elsewhere where previously Vibrio could not persist. And so there's been a lot of work and including in the marine environment, just talking about how climate change is going to really shift this host pathogen environment equilibrium. We expect that these changing environments will drive animals into new areas, that they'll interact in ways that could facilitate disease transmission in a way that it has not happened before. We really worry about emerging infectious diseases in this realm. We know that increased connectivity, whether that's through loss of sea ice or animal movements, that will increase the pace for new pathogen sharing and emerging infectious diseases. And so there's major concerns for populations that are sensitive in the California coast and elsewhere. And I try to show this figure of this iceberg because I feel like we just know absolutely so little about especially emerging viruses. We have a couple, few cases, scattered cases that have been done by the great work of many veterinarians across the world working on marine species. But really that's where we see die-offs or in animals that are able to be studied because they bring their prey and exist on the surface. So we have huge knowledge gaps around what viruses are where, what species are major components of transmission. And I think we need to think, of course, beyond just marine birds, certainly within our marine mammals, certainly with influenza. There's marine birds and marine mammals and lots of other species in the mix that are creating concerns. And of course, the specter of high path, even influenza, I'm sure will be discussed, but that is a really important concern from work that's being done elsewhere in especially Latin America. So something that we're expecting to be an issue in the near term for us here in the US. And I think, thank goodness, the work that the marine mammal community has done over time has really fostered a lot of the transitions in thinking, and out on the West Coast bolstered our opportunity to really push forward this idea around One Health and how important even human health is on ecosystem integrity. And I think the work that was done around sea otters and how reflective they are of changes in the nearshore ecosystem really was able to for many of us, a huge contingency of researchers and agencies working, tying health of sea otters to the ecosystem and support that we got through NOAA and the oceans early on, very visionary Oceans and Human Health Program, to really make the case for environmental change and the impacts that has on indicator species such as sea otters, but also for humans who share that same system and eat many of the same prey as sea otters. So these transitions are happening globally. This is an example of work that is being promoted by the United Nations around transitions to One Health and sustainable development that include fisheries and oceans, of course. And so I have huge acknowledgements. None of this work that I presented is my own, almost all entirely done through these amazing agencies and leadership and vision done by state and federal agencies that have participated in sea otter recovery and pushed the species forward to where it exists today, and continues to monitor and do just absolutely phenomenal research and science to be able to make a case for this. Thank you so much.
Thank you very much, Chris, for a very interesting talk. Again, bringing to point the complexities of trying to do conservation on these animals. Our next speaker will be Jonathan Sleeman, of the Science Advisor for Wildlife Health of the USGS, talking about surveillance and management strategies to address climate change and wildlife health. And I don't know why, but it just does not
It's an
respond.
extended display, so we just have to keep moving it this way.
Thank you. Thank you.
Good morning, everybody. Thank you very much for the opportunity to be here. It really is a privilege to see everyone. So I mostly work on terrestrial ecosystems and terrestrial landscapes. So I want to make that apology right up front, but I'm hoping a lot of what I talk about is very germane to the work in marine ecosystems. And actually, it's been very pleasing to see a lot of the concepts I'm going to talk about have already been discussed. So as you probably know, we're very much aware that climate change is having profound impacts on biodiversity. Changes in habitat availability, we've heard, food availability, migration patterns, phenology. And the assessments of global warming have shown that if we reach a 2 degrees Celsius rise in average global temperature, we're going to see probably an 18% decline in insect species, 16% of plants, a whopping 99% of corals will go extinct, and 8% of vertebrates. So it really is a serious crisis. We're also, as we've also seen this morning, seen significant evidence of climate change effects on the incidence and geographic and severity of a wide variety of fish and wildlife pathogens. And just an illustration, there's a study came out recently that showed that global warming is going to influence animal migration patterns, bringing humans, animals, livestock into closer contact, increasing those opportunities for spillover events and potential epidemics and pandemics. So as a consequence of this, a group of us in USGS organized a multi-agency, multi-partner workshop back in January of 2020, just before the pandemic. It feels like a lifetime ago, where we sort of got together, sort of experts, managers, to come together to sort of talk about this problem of how we're going to address climate change and wildlife health. I want to talk about the 4 key recommendations that came out of the workshop, which is, as you heard from Francis, the need for systematic health surveillance, the idea of taking a systems approach to these problems, the concept of managing for health and not disease, and finally, the implementation of climate adaptation strategies, or so-called nature-based solutions. So as Francis mentioned, we are very much working in the dark in this problem. The lack of long-term systematically collected data is an impediment to our understanding of the effects of climate change on wildlife health. The systems where we have that long-term data, we have a very good understanding of the impacts. The systems where we lack that data, we have no understanding. And a very nice example of this, and it's somewhat serendipitous, was there was a long-term monitoring program for abalone off the coast of California. And they went out and collected biotic and abiotic data on a regular basis over time. And they started noticing a number of these abalone were turning up with this, what they named withering disease, which obviously is shrinkage of the foot. And because they had that long-term data set, they're able to find that it was correlated to ocean water temperature and then elucidate the bacterial cause of this infection. In fact, to go on to show that different species of mollusks were differently susceptible to this effects of temperature. So the question is, why don't we do a better job of wildlife health surveillance? Well, I think that the challenge is it's quite complex and quite expensive. In order to have a sort of robust wildlife health surveillance system, you need a network of partners in the field that can detect morbidity, mortality of wildlife, who can go out and collect samples for you in the field data. You need the diagnostic laboratories that have the expertise and the ability to deal with wildlife pathogens. You need to have some sort of system for managing, storing that data with all the data standards. Then you need the ability then to sort of relay that information, analyze that information, and relay it to decision makers who can then take action in the field. And I think very much our wildlife health surveillance systems in the United States and probably elsewhere are very much imperfect. I think a nice illustration is the fact that I think that NOAA has a very well-established, formalized network of partners, the Marine Mammal Health and Stranding Response Network, which is situated across the coast, through cooperative agreements, is able to contribute to a national network. For terrestrial systems, we do not have the same type of network. We rely very much on the voluntary participation of our partners, which are the state and federal wildlife management agencies. And I think on the flip side, I think we have probably a sort of more advanced centralized laboratory and diagnostic system with the National Wildlife Health Center, with the expertise under one roof that can do the wildlife diagnostics and data analysis. And we're also, as I'll show in a minute, working on hopefully a more national level standardized data storage and standard system. But so maybe one of the things for discussion is how can we leverage these different expertise and capabilities to create a more robust wildlife health, marine and wildlife health system for the United States. So again, in recognition of the fact that our data systems were lacking, it was about 10 years ago we got together partners and decided to try and create a more nationalized data information management system for wildlife health. We called it WHISPERS. You have to have a cute acronym, right? It's government. The Wildlife Health Information Sharing Partnership. And it's basically a centralized repository where people put data and information about wildlife mortality events. You go to the homepage, you've got a very simple map. If you click on a dot, you can see some summary information about that mortality event. And it provides very important situational awareness regarding what's happening in the country at the current time. But you can search by historical data, you can search by species, disease, geographic location, and obviously help to understand a little bit better some of the incidences of disease events. I can tell you that it took a lot of very unglamorous work to get this off the ground. You have to do all the case definitions, you have to do all the sort of data standards and data harmonization, the data dictionaries. A lot of sort of background work goes into establishment of, and it's one of the challenges. Also, getting people to share their data is a challenge. People are very concerned about sort of loss of data privacy and data integrity, their data being used for reasons they weren't expecting. You have to make it easy for people to use the system, and they have to see a benefit from participating. And perhaps in the panel session, we can discuss a little bit more in depth some of the strategies we use to encourage data sharing. But we do have ambitious plans now to move beyond just mortality data to actually collect more systematically, targeted or active surveillance data, as well as potentially integrate other data sets like the environmental data that Francis alluded to. And I think if there's no better argument for the need for data integration is avian influenza. Clearly what happens on the terrestrial landscapes has an impact on the seascapes. And obviously right now we're seeing some pretty profound mortality events as a consequence of highly pathogenic influenza, particularly in Latin America. This is a picture of the southern elephant seal die-off in Argentina, where there's estimated 100% pup mortality, which is clearly going to have a profound impact on population recruitment. So moving on to the kind of more of the interventions and managements, I think one of the challenges with climate change is a wicked problem, right? The problem's long-standing and attempts to solve it have failed. it was simple, we'd have fixed it by now. There are multiple perspectives on the cause of the problem, what should be done. You've got diverse stakeholders. They find it difficult to align their objectives and their efforts. There may be even competing objectives. And actions may have unintended consequences, probably because you haven't necessarily included all the potential stakeholders. So one of the recommendations from this workshop is really to take a systems approach to addressing these issues. And actually, you saw some examples, I think, of very interesting systems in marine environments. The idea is that the concept of the iceberg model is what you see is the actual events. So it could be a mortality. But what actually drives those events are the patterns of human behavior or the trends over time. What drives that behavior is the structure of the systems, the economic incentives, the rules, the regulations. But ultimately, what drives that system is sort of human values, in mental models and cultural preferences. So, for example, who'd have thought marine mammal mortality in South America will be a consequence of farming systems in East Asia, which is a consequence of food preferences, the preference for live animal markets and freshly slaughtered animals, which is a deeply held cultural practice in China. So the idea being is by understanding the sort of patterns of behavior and the supporting structures and mental models, it may allow you to sort of get new perspectives on the problem and to identify potentially new leverage points in the system. I'm going to use a very sort of simple example to illustrate this. You can also kind of map these systems through like causal loop diagrams. It just provides you a visualization of your system and how things that are causally interrelated. This is a very simple diagram of a sort of positive feedback loop. So I'm a chicken farmer, and those chickens lay eggs, which leads to more chickens. But unfortunately, on my farm, there's a road, and the chickens, for some reason, want to cross that road, and that leads to loss of chickens. And so how do I, as a farmer, increase the number of chickens in this system? Well, I could improve the food, the husbandry, increase the number of chickens, increase the stock in the system, but that costs me money. I could fence the road, but that costs me money too. Fences fail. I could close the road, but maybe that road's to the best whale viewing site in Hawaii, and people are going to get angry that they can't view the whales. But ultimately, if I really wanted to solve this system, I need to convince the chicken not to cross the road. Now, I know you think I've gone mad, but the point is I need to understand why that chicken's crossing the road. Well, maybe there's good habitat on the other side of that road, ideal food source that I could replicate on the other side of the road and actually prevent them from crossing the road in the first place. So my point is, with this very silly example is the idea is you've got a system, and within those systems, there's different leverage points. You can change the parameters of the system, change the stock by adding eggs. You can change the feedback loops by fencing. You can change the design of the system by closing the roads, or you can ultimately focus on the sort of mental models and convince the chickens they don't need to cross the road in the first place. The point is the higher up you go in that leverage is the more impact you could potentially have. Now, you don't have roads in oceans, fortunately, but you have shipping lanes. So maybe one example that I could think of is some of the boat strikes like in manatees, and maybe mapping that system, understanding why manatees and humans are using the same ocean at the same time could help identify some potential interventions and leverage points. And here's just a very good example. It's a climate change example, but a terrestrial example of using this systems approach. So when ocean temperatures rise due to El Nino events, this leads to droughts in eastern Australia. This causes food shortages for fruit bats in Australia. And what happens then, those fruit bats then move to urban areas to forage for food. These fruit bats carry a virus called Hendra, which this causes high mortality in horses and people. And a consequence of those bats moving into human areas, we start to see outbreaks of Hendra in horses and people. So how am I to intervene in the system? Well, I could kill the bats, solve the problem. I could potentially discourage them from using human areas, exclusion devices. I could vaccinate the horses, but really the win-win solution is reforestation. If I build and create new habitat for these bats, I decrease their need to move into human areas. I preserve the bats and the ecosystems they provide, and also protect human health. So it's led us to develop this sort of concept of the One Health impact pyramid. The idea being that there's different levels of intervention, starting off with simple education to a clinical intervention, long-lasting protective measures like vaccination, changing the context to make decisions healthy, which is laws and regulations and policies. But really, sort of the true determinants of all health are the socioeconomic and the environmental factors. So we focus our interventions on those two bases of the pyramid. We're going to have much more increasing impact and potentially find those win-win solutions. Trade-off is that it's much more effort to do that. And so there's obviously going to be a trade-off between effort and increasing impact. The next concept that we discussed is the concept of managing for health and not disease. We recognize, and I think we had examples of this morning, that health of wildlife populations goes beyond the absence of infectious diseases, contaminants, and includes population sustainability and resilience. And because we can't control the climate, and sometimes we can't even manage the disease, maybe focusing on non-climatic stresses, such as pollution, overexploitation, habitat loss and fragmentation, invasive alien species, to increase the resilience of these populations may actually help us to find interventions or improve the resilience of these populations so they can thrive even in the face of some of these disease outbreaks. Again, recognizing that not all ecosystems can be managed or there are priorities that I think the National Park Service developed this what we call a RAD framework to help us sort of figure out which systems require which types of interventions. The idea being that for some systems that are very important, you resist that change, try and maintain current or historical ecosystem function. There may be some systems where you want to change it, but you can actively steer that transformation towards a particular ecosystem structure or function. And there's some ecosystems where we may just have to accept those changes and not intervene and focus our efforts and resources on those where we can potentially have an effect. And I think, you know, from a marine mammal perspective, I think health promotion is an especially important concept in these ecosystems. In preparing for this presentation, I found this very nice paper where they reviewed strategies for managing marine disease and looked at the sort of applicability in marine ecosystems. And what they found is a lot of the tools we have in the terrestrial system, biosecurity measures, quarantine, population manipulation, vaccination, all those very nice examples out here today, are yet less applicable in marine environments. But things like biodiversity, habitat conservation, laws and policies like endangered species list can be. And I think it probably places an increased emphasis on those types of interventions, enhancing protective areas, looking at rules and regulations as a way to manage these problems versus the classical veterinary preventive measures that we often use in terrestrial systems. And then finally, the kind of last recommendation was look at these climate adaptation strategies, these ecosystem-based or so-called green or blue infrastructures that provide services that help people adapt to the reverse climate change, but also provide wildlife habitat. And the classic example here is a sort of restoration of coastal mangroves and coastal wetlands, which provides a buffer to the human communities when hurricanes hit the land to actually reduce the impact of those hurricanes, but also provides very nice habitat for wildlife and coastal species. I'm not aware of many sort of nature-based solutions in the ocean, but that's something for increased discussion, maybe like kelp forest restoration, seagrass restoration. Maybe that we can open up a whole sort of deep blue infrastructure concept. So finally, just summarize kind of the key four recommendations. We need long-term systematic surveillance to identify impacts, as well as assess our management actions. Using a systems approach to help identify leverage points for action. Looking at health promotion strategies, reducing those non-climatic stresses such as pollution, overexploitation, and so forth. And looking at these nature-based solutions, the wider adoption of conservation and sustainable use practices, including the strengthening of protected network areas. So thank you very much. Thank you.
Very interesting. Okay. Our next presentation will be from Kim Damon Randall, who's the director of the NMFS Office of Protected Resources. And she'll be speaking to us about the NMFS Marine Mammal Health and Stranding Response Program at 30, statutory mandates and future directions in a changing climate. It doesn't look any older than 25.
So thanks so much for the invitation to be part of this panel on marine mammal health in a changing climate. I'm really happy to be here to speak on behalf of the NMFS Office of Protected Resources about our agency's investment in monitoring and understanding the health of wild marine mammals and how climate change will continue to present us with challenges and opportunities. So prior to the MMPA becoming law in 1972, a lot of work surrounding marine mammal strandings had already occurred. Museums around the world were interested in retrieving stranded animals for their collections, with the largest effort actually taking place here in Washington, D.C. From the early 1970s through 1992, the Smithsonian Institution worked with their staff and volunteers to build a network of partners along the eastern coast of the U.S. to respond to strandings. While there were other stranding efforts in the U.S., they were fairly localized. And in 1992, Congress amended the MMPA to include Title IV, which created the Marine Mammal Health and Stranding Response Program, or MMHSRP. This new program was an explicit health mandate, which is pretty rare within my agency, and worked over the last 30 years to promote investigations into marine mammal health. 3 events happened within a relatively short amount of time in the late 1980s to trigger the creation of this program. There were multiple humpback whale mortalities in 1987 due to biotoxin, a bottlenose dolphin die-off in 1987 and 1998 from a morbillivirus, which resulted in a better understanding that dolphins had very high levels of contaminants in their body, and the Exxon Valdez oil spill, which occurred in 1989. These events represent the wide range of threats that are faced by marine mammals in the wild and are sources of injury, illness, and mortality that we still see today. In Title IV, Congress sets out 3 statutory purposes of the program. The MMHSRP was established to facilitate the collection and dissemination of reference data on health of marine mammals and health trends of marine mammal populations in the wild. Correlate the health of marine mammals with available data on physical, chemical, and biological environmental parameters, and conduct effective responses to marine mammal unusual mortality events. Collectively, these show a need to understand health threats in the broader context of the marine environment, as well as the congressional direction to consider health in a broader sense, not just through strandings. Old school with paper here. Over the last 30 years, the MMHSRP has significantly developed a broad program to meet these mandates. Our efforts today focus on 4 primary areas within marine mammal health: coordination of the marine mammal stranding and entanglement response networks, including collecting and managing the relevant data from these response efforts and using them to inform management, leading coordinated and effective response and investigations into unusual mortality events, or UMEs, conducting and supporting biosurveillance and baseline health research on wild marine mammals using a variety of data collection opportunities and partnerships to understand sublethal and lethal health concerns and trends, and supporting response networks and researchers on marine mammal health through the administration of funding sources, including the Prescott Grant Program, which was created with an additional MMPA amendment in 2000. One of the most direct ways that the MMHSRP helps inform management is by understanding human-caused threats to their health and welfare. The program collects and summarizes data on interactions with fisheries, vessel strikes, marine debris, and deliberate human harm, including gunshots. The MMHSRP has worked with partners to develop and implement a standardized form of collecting information from strandings across the United States, creating reference data to identify and monitor hotspots and trends. We help support a process for the documentation and analysis of recovered gear from entangled animals to identify issues with particular types or configurations of gear. Findings provide the basis and support for Office of Law Enforcement investigations, take reduction teams, and NMFS management efforts. We aim to provide the information to help managers identify changes that can help reduce the threat and benefit the marine mammals while still allowing human activities like fishing and boating to occur. This standardized information is also key to identifying when adopted management measures are working through a reduction in observed human interactions.
The newest changes are a result of the Marine Mammal Research and Response Act, which was enacted in December, just a little under a year ago, as part of the National Defense Authorization Act of 2022. It amends Title IV of the MMPA to create Health Map, which is an online database to store additional information on health and visualize information on IUCE. Require the stranding network to submit additional health information to Health Map. Historically, we have required basic information on stranding events to be collected and shared with NMFS in a standardized way. These new mandates will drastically increase the amount of information we will be standardizing and collecting. And it includes amendments to the Prescott program and the creation of the DRAWC Rapid Response Fund. The MMRRA will create an expanded standardized database called Health Map, which I mentioned. This leads to enhanced data accessibility by ensuring that Health Map includes supplemental data on stranding events and laboratory analysis results, including virology, bacteriology, and parasites. It will result in a better understanding of the causes of stranding and causes of death for animals that were found deceased, and identification of additional threats beyond human interaction, including biotoxins, contaminants, and infectious diseases. We have to also recognize that these new opportunities do come with some costs. Reporting the additional information expends time and effort by the network partners. While the new language does not require additional analyses beyond those that are already collected, there may be some additional needs as this mandate is implemented. Finally, NMFS will have to make additional investments in developing, maintaining, and validating the Health Map database. One way to think of the stranding and entanglement response networks is as a nationwide monitoring effort, providing NMFS with high-quality information on a broader geographic scale. In addition to working with stranded and entangled animals, out of habitat and visibly injured cases, the MMHSRP also conducts and supports baseline health research on free-swimming animals to investigate their health status. We work with a network of collaborators on live animal health studies. In some cases, this is following a known threat such as an oil spill or disease outbreak. We also collaborate with additional sources of information on marine mammal health, including subsistence hunters in Alaska and animals obtained as fishery bycatch. Together, the live and dead monitoring contributes to and sometimes represents the only best available science that we have for management and science efforts within NMFS, including for take reduction teams, serious injury determinations, stock assessment reports under the MMPA, recovery planning and biological opinions under the ESA, permitting under the MMPA, ESA, and other agency mandates, and natural resource damage assessments, which are conducted after an exposure event. Sorry, lost track of my order. Outside dollars for all stranding response nationwide are privately raised, except for the $4 million that we have in Prescott grants. There is some federal support through the Prescott program, about $4 million historically, but recently the authorization level, not the allocated level, important distinction there, has been raised to $6 million. However, we recognize that these funds, while impactful, do not fully cover the cost for response, rehabilitation, and release. Therefore, much of the work must be privately funded through resources raised by our partners in the response networks. Other successful partnerships include NIST, which oversees the National Marine Tissue Bank, which is the primary long-term archive of marine mammal tissues for retrospective analyses in the United States. The National Wildlife Health Center, which we have recently partnered with to provide some histopathology and diagnostic support for cases of stranded marine mammals, and we are interested in exploring ways to expand that partnership. And permit collaborators, including partners in academia and NGOs, with funding support from the Office of Naval Research, the Marine Mammal Commission, and many others. Marine mammals are an important indicator of species, important indicator species of ocean health, as we've been talking about. They are top-level predators that eat many of the same fish that we do, and many species live in coastal areas that are utilized by people. When marine mammals show signs of illness, they may be signaling changes in the marine environment that might have significant implications for the overall health of our ecosystems. Climate change will result in many ecosystem changes, which will impact marine mammal health, reproduction, survival, and recovery. The monitoring of stranding and entanglement response networks and investigations into marine mammal health can help detect even minor changes. This will require additional resources to fully investigate these questions. So, thank you.
Thank you very much, Kim. And Happy anniversary to all you MMHSRP folks in the back, the troublemaking row, second down from the top. Okay. Is there just a minute to go back here? I can make it reappear. You do. Our next speaker is the Marine Mammal Commission's own Scientific Program Director, Lori Schwacke. And she'll be talking to us today about a vision for a nationwide marine mammal health monitoring and surveillance plan in a changing climate. Thank you.
Thanks, Randy, and thank you all for being here today. As Randy said, I'm going to talk about a vision for a nationwide marine mammal health monitoring and surveillance plan. It's not my vision. This has been the vision of many folks within the commission and within NMFS. You've heard a lot about the history here today. For a long time. The vision is to be able to detect changes in population health before we see population declines and before potentially we can even detect changes in vital rates. The idea that we can get early warning so that we can respond better and come up with better plans for response. What I'm really going to be talking about today is recent activities and specifically a workshop that the commission held last spring to pull together participants and talk about what the surveillance plan would look like moving forward. The errors not work. Oh, there we go. Okay. So why do we need a surveillance plan? And you've seen this slide before. It was in Francis's, and it's the same as what don't we know? And that's why we need a surveillance plan. We need data associating health changes with environmental measurements, which are scant. And I would say not only are the associations with environmental measurements scant, but just population health data are scant, other than a few limited species. This hinders early detection of changes in population health. Also, the distribution of infectious pathogens in marine mammals is largely unknown, as others have pointed out, due to large-scale systematic sampling for specific pathogens over time being so rare. This, again, makes prediction and response preparation for disease outbreaks difficult. We need to be proactive rather than reactive. We want to be able to detect these things before they happen, so that we have time to plan for response and hopefully design interventions or mitigation strategies. As others have described, the cascade of environmental changes from changing climate will be complex, and we need to be armed with a solid understanding of population health and an understanding of what the physical, biological, and chemical factors are that will affect population health in order to effectively respond. Kind of the systems approach that Jonathan was mentioning. Again, from Francis's talk, who stole it from Sue, the marine mammal health monitoring and surveillance is a critical step in the path towards using marine mammals as ecosystem sentinels. It's essential for NOAA's One Health goals and will rely on and be an important part of the global ocean observing system. So before going into the plan, I'd like to clarify some terms, and specifically what is the difference between health monitoring and disease surveillance. We talked a lot about this at our workshop. Monitoring is ongoing efforts, systematic efforts to assess the health and disease status of a given population. It's continuous and systematic over time and includes routine observations of health and endemic disease prevalence, as well as observations of environmental factors. Surveillance, on the other hand, implies some form of directed interaction will be taken if data indicate disease above a given level. It looks for emerging disease. It can look for spread of disease to new areas or species and populations. It'll become clear as I talk through our goals for the plan that we need both here. We need both monitoring and surveillance. From here out, I may just refer to the surveillance plan. I mean monitoring and surveillance. I don't want that to drop. So as I mentioned, what I'm presenting here are outcomes from a workshop that was held last April. The purpose of the workshop was to develop a national plan for temporally and spatially structured marine mammal health monitoring and surveillance. It included veterinarians, epidemiologists, statisticians, HAB researchers from across NOAA, the Fish and Wildlife Service, National Wildlife Health Center, and other subject matter experts from academia and non-governmental organizations. The goals of our workshop were to review primary objectives and components of a monitoring system, to prioritize exposure parameters, species, and populations to be monitored, and to discuss potential approaches for conducting the monitoring and surveillance and outline a sampling plan. I want to recognize the participants here that were involved in the workshop, and you'll note that two of these participants, Michelle and Chris Johnson, are also speakers here today. So we thank them very much for their commitment and their willingness to devote so much of their time to help push this vision forward. So in the workshop, we engaged participants in discussions covering three primary topical areas: infectious disease, harmful algal blooms, and then non-infectious disease or health endpoints that are of interest. Again, we don't want to let that drop. The monitoring of population health is very important. For infectious disease, we organized it by viruses, bacteria, fungi, protozoa, and parasites. For HABs, some of the priorities were domoic acid and saxitoxin, especially on the West Coast and the Northeast, as you've heard from other talks today. Brevetoxin in the Gulf of Mexico, which I'm sure Randy can tell you all about because he deals with it very frequently. And then microcystins, which haven't been such a big issue for marine mammals yet, other than maybe sea otters. But with changing climate and increased freshwater inputs from terrestrial sources, we do worry about microcystins, which are a freshwater algae or produced by freshwater algae, becoming more of an issue. Speaking of freshwater, of the non-infectious disease, we worry about freshwater disease, particularly in coastal species, bottlenose dolphins in the Gulf of Mexico. For large whales, we think about body condition as a priority because that may be how we detect changes in health related to shifting prey distributions or changes in prey quality, some of the talks that you heard yesterday. And then, of course, other pathologies, cancer that's been related in Francis's work with contaminants on the West Coast. This list is not exhaustive. The workshop participants discussed a number of other health markers that can be predictive of reduced survival or fecundity as well. So we identified three criteria for prioritizing the pathogens, biotoxins, and health outcomes for monitoring. First, potential for population impact. We talked about, what factors might cause high mortality or long-term reproductive effects. So populations may be able to sustain one bad pupping year or calving year, but having disease that affects reproduction long-term is a much bigger issue from a population level. This is even more important if the marine mammal species has a small population size or is of critical conservation concern. We talked about pathogens or biotoxins that may be climate sensitive, so they may increase with weather temperatures or changes in water chemistry. And we talked about zoonotic potential or importance as an eco-health sentinel. And Chris talked about that some today too. We worry about potential transmission to other species, including humans, and whether the detection of the agent could inform risk to other species within the ecosystem. Again, this is more important if there are other species that are threatened or small populations, which you may not be able to detect the agent. We maybe could detect it in other populations within the same ecosystem. And we talked about different sampling approaches. So we've heard a lot about strandings this morning. There's also subsistence harvests and bycatch, and then targeted research sampling. And as you go down this list, you kind of have more control over the actual sampling design. With strandings, you're somewhat limited in that you can pick how much to sample or what you're going to sample for and how many animals to sample, but you're kind of limited to what washes ashore and what you can get. And often that's young of the year, or they're ill or malnourished animals, so they're not necessarily representative of the overall population. And sampling is generally guided by case investigations in these cases. Subsistence harvest and bycatch offer somewhat controlled or more control. It's generally random sampling of free swimming animals, so maybe more representative of the population as a whole. And you can select from specific species or demographic groups, but it's all based on what the hunt produces. And then there's targeted research sampling, which of course you have the most control, either temporary capture of animals that Michelle spoke about earlier with monk seals, remote sampling, either biopsy or other remote sampling techniques, and then visual health assessment or photogrammetry. This can be random in principle, but as we all know, there's always some biases in sampling. Biopsy sampling, you tend to, at least with dolphins, you get more males than females. So that has to be considered when you're doing the sampling. Okay, so using all this information from the discussion of the priority pathogens and toxins and health indicators, we considered available sampling approaches, and we tried to identify some priority focal species based on the value for monitoring and the available samples for monitoring. So we put this on a graph with value for monitoring on the y-axis and available options for sampling on the x-axis. And we walked through with these criteria for the value of monitoring. We looked at management priority, specifically things that were ESA listed or depleted. We looked at ecological importance, such as the importance of sea otters in their ecosystems. We looked at likelihood of being affected by a changing climate and exposure to multiple stressors. So we all know many populations are exposed to multiple stressors, and the more there are, the more opportunities there are for interactions that are going to produce results that we don't anticipate, these ecological surprises. We talked about available options for sampling, looking at feasibility for hands-on sampling being kind of the creme de la creme, remote sampling options, sampling from subsistence, harvest, or bycatch, and then the carcass recovery rate. I mean, basically some animals are going to strand because they're coastal species, others you will never get the carcass. So looking at all this, we tried to just organize species based on their value for sampling and their options for sampling. I'm showing the example for odontocetes, but we did similar plots for other taxa groups, mysticetes and pinnipeds, and also discussions of the fish and wildlife species, polar bears, sea otters, and manatees. And this is a map showing the what we came up with for cetacean focal species along the coast, so covering the entire nation. We put this all together considering regional concerns and priorities, and identified the focal species for the US coast and offshore waters. This is the example with cetaceans. And here we have our map with the pinniped focal species. And if you're looking at this and saying, oh, that's almost every pinniped species, yes, you're right. That was pointed out to me as we were putting these slides together. And the thing is, though, pinnipeds do represent different habitats. Some are near shore, some are offshore. While there are a lot of them up there, they do all meet important monitoring or surveillance needs. But obviously, we need to go back and give some more consideration of, maybe it's not a yes, no, focal, non-focal. Maybe it's more gradient of importance for sampling. It was pointed out that hooded and harp seals aren't up there. And then somebody chimed in and said, oh, you need to put hooded and harp seals up there. So it will be exhaustive. So using all this, we created tables of priorities by regions and species or taxonomic groups for the pathogens and toxins and the some of the non-infectious disease endpoints as well. So I know you can't read much of this table. Is still under review by the participants as well, so it still may change. What I'm showing is just an excerpt from the Southeast US and the Pacific Islands, because those would fit on the slide. And the Southeast doesn't have pinnipeds, which is why it fits on the slide. But we have developed similar tables for the Northeast, West Coast, Alaska, and Pacific Islands. Okay, next steps. We plan to publish the workshop report online, hopefully soon. Either the end of this year or early in 2024, January timeframe. We're hoping that this online report is going to provide a resource for field biologists and researchers to prioritize their sample collection and analyses, and that make those samples and analyses contribute to a broader nationwide health monitoring surveillance to detect climate change impacts. We then will distribute the plan, meet with agencies and organizations, and encourage funders to consider projects that could contribute to this national marine mammal health surveillance. And then an implementation plan needs to be developed, including details of the sampling plan. Who are the partners? You know, how getting more into the details of how frequently do we sample, you know, what can be produced. This is something that NOAA would obviously lead, but the commission would certainly be happy to support on that. And then begin to build capacity with existing resources through partnerships. And this has already been discussed. Kim talked about Health Map. That's certainly a step forward towards the information management, which was one of the 4 components that Jonathan talked about in the components of a surveillance system. So that's already happening. Obviously, the stranding networks are already partnerships, but we need additional partnerships to expand that beyond just strandings. And then finally, we'd like to formulate some recommendations for building additional capacity that is necessary to fully implement the components of the plan. We assume that this may be iterative, building over time, but we've got to start. And with that, I would like to thank the workshop participants and presenters at that workshop as well. And also thanks to all the folks from the commission and NMFS who helped with putting this presentation together. Thank you.
Thank you very much, Lori. I would also like to thank the folks who helped put this session together. From the commission, Lori Schwacke, Tiff Brookins, Lori Leach, and Patty Roselle were all instrumental in helping to bring the speakers together for a very, very interesting set of presentations. We're running a bit late. Fearless leader, how would you like to proceed at this point? It's supposed to start at 10:45, I think. 20-minute break, which brings us back at, I don't have a 10:50 back in the room for the panel discussion. Thank you all very much for your attention.
Thanks, honey. I'm just out there.
I'll say that one today, maybe because I'm saying two things are going to
No.
A hush
No,
falls
this-
over the crowd.
So I turned it around.
Dr. Schwack?
Wait, it's not-
Oh, there she is. Okay. Thank you all for coming back promptly. We want to have as much time for this session as possible. And we're going to deviate a little bit from the protocol that we had yesterday. For an old guy like me, the writing is too small up there to see, so I can't read it. Actually, there's been a request from one of the panelists who is in a position of power that we do things a little bit differently this time around. Well, what I would like to do first is ask the commissioners if they have any questions. Dr. Reed.
Thanks, Randy. And again, thanks everybody for a really good, yet another good panel. I learned a lot this morning. It was really good. My question is to Michelle. And as you know, I'm a huge fan of the Monk Seal program. I'm just so impressed with what you guys have accomplished. I was thinking about the vaccination program and the factors that made that uniquely possible with monk seals. Small population, and the fact that from your observational program, you know almost every individual, you know, relationships that allowed Jason to do the modeling work he did. But I was trying to think about, as we think about that as a model for other species, presumably we'd only apply to other populations that are small, because otherwise it would be not feasible. But my question is, if you only knew half of the animals, or you only knew a third of the animals individually, could you still develop an effective program? Because there are going to be so few populations for which we have the depth of knowledge at the individual basis that you do for monk seals.
That's a great question, Andy. And I think the answer is yes. You know, we do have the luxury of knowing our individual seals pretty well. So some of the things that we talked about when we were figuring out how to implement this vaccination strategy was, do we need to mark them at the same time we're vaccinating them so that we don't vaccinate the same individual by accident 6 times and then miss other individuals in the population? But I think depending on the species you're talking about and the different resources that you have and the logistics of those studies, I think there is potential to figure out how you can mark or identify those individuals. Is one of the things that we need to think about in any vaccination program, whether we're talking about terrestrial or marine wildlife, is that we want to be able to go back and understand if that disease shows up, was it from the disease showing up, or was that seal or animal vaccinated? And so we got around that because we're really fortunate that we've got this small population where most of those seals are flipper tagged and otherwise identifiable. But natural markings can be a help. So if you're thinking about bottlenose dolphins, they have distinctive fin markings. So there's a lot of successful photo ID. And as technology changes, I think we'll have increasing capacity to identify those individuals. So I do think it is possible to implement. And I think the other thing that comes to mind is herd immunity and thinking about, do you need to vaccinate every animal, or can you vaccinate a subset of that population and still dampen or minimize the effects of an outbreak? And that's going to obviously be species dependent, but I think it's something to look at carefully, species by species.
Yeah. Follow up, John.
Sorry, if I just add a few things. One thing to consider is there's a lot of advances in vaccinology happening right now. And one of the concepts is these propagated vaccines, which means when you vaccinate an individual, that individual sheds that vaccine, which will then go and infect another individual. So it may be possible in the future to only vaccinate a subset of the animals, yet still get enough herd immunity. I think these may be a few years off, but there's a lot of exciting opportunities with these new technologies. Thanks.
I was going to add one other thing too, is that in some wildlife systems, you can put a marker in the vaccine. So, so you don't know who's vaccinated, but if that animal should die, there is a marker that indicates that it was vaccinated, like a sort of tracer.
I'm going to take a much broader approach and not get into vaccinations at all. I'm really thankful to all of you for your presentations. I was really looking forward to this session. so that Francis and I can stop talking about that simplified arrow diagram and take it a bit further. I'm really pleased to see some of the outcomes from the surveillance workshop and that you have that regional approach, because it lights up my brain in terms of IOS and the regional framework that IOS has. We talk a lot about HABs, and I think for marine mammals, that might be a low-hanging fruit that we start to develop these connections for health map around. But I also wanted to put in people's minds, if it's not enough already, is heat and ocean heat events and storm events, and how I think actually Weather Service could be very helpful in this regard too. I'm curious about Weather Service and whether it was part of your scheme that you showed us with the mapping, but we can talk about that later. I don't really want to put anybody on the spot, but if I would put Lori and Carl at either end of the table for just your thoughts about how we might, excuse me, go from the great endpoints that it looks like your workshop is at and link that into what is already in development or available through the regional associations of IOs, sorry, especially with regard to heat and HABs, but just your thoughts on that at this juncture.
I can go first. I'll throw you under the bus. Because
well, that doesn't sound good.
I briefly talked to Carl yesterday after the meeting, because I was excited, to hear in Clarista's talk about how she was integrating with the stranding network on the West Coast. And it was almost a plea, it sounded like, from her to make this more of a formal process. And I think that needs to happen. And I think it would help to have some consistent, I understand there's regional needs and differences between the regions, but if we can look at things from a national perspective too, and have some consistency, and especially if it's just like, how do you communicate between the stranding networks and the IU's folks? Throw it to you.
Yeah. So you put me and Kim next to each other, which is good. I was just in the break. So let me tell you a little about how within the IU's network, we have this great legislation that creates this regional to national structure. There's a lot of capabilities built from the ground up, bottom up, developed. Individual principal investigators figure something out, they start to do it, we start to implement it. And we have ability through our grant making mechanisms to have top-down instructions as well. And we sort of try to work along that spectrum and figure those things out over time. And we build communities of practice around different technologies and application areas and decision support tools and things like that. So the Animal Telemetry Network, Marine Life Program, I've been pushing biologists for years to help me identify what we can do operationally. And so this is an example of that maturing and what we can potentially do. And with that, we run a lot of ocean acidification sensors through our network. The Ocean Acidification Program is the expert on that. We do a lot of HAB work. The regional experts like Clarissa and then the National Centers for Coastal Ocean Science are the science experts within NOS for that. And so Kim's team is the expert on the health network. And so her guidance to me about what would work nationally is the thing I would look for with you all. And MMC can push us to do what makes sense and try to push us faster. And I would accept that pushing. I may not be able to do it as fast as you like, but I'll listen and I'll say noted, and we'll try to add it to the list. And Sam Simmons did a detail in our office a few years ago. So Aaron Lebrecht and I and Sam were talking about repeating that behavior again. And Sam was reminding me that the distinction between the regional delivered capabilities versus national, and can we find a solution that is consistent across all? And the answer is yes, and we should be able to do that. And so developing the conversation to the point where it will work in these ways with the differences based on your workshop. I love seeing that, because that's a list of the requirements for what would work in a given region. And you use that with our regional experts, figure out what the communications networks need to be and the participants and those things, so that there's functionality there. So all of that logically makes sense to me. And finding a solution technology wise to make the environmental data and the health data integrated. We've got multiple instances of doing that in the last 5 years. And so I think Kim's team's been making good progress on that. And there's next steps that are identifiable, and we can keep moving that forward.
I'm really encouraged very much by the results coming out of your workshop, and I know it was a big effort by all. But again, I'm agreeing and shaking my head yes with everything you're saying, because that's my experience of working with several of the regional OOSs, that this is doable. So what we need to do now is the thinking, and maybe the systematic thinking of where the inflection points are and things like that. So got a lot out of your presentation as well. So thank you all.
Andy, any other questions? Okay. And Francis, do you wish to enter in as a commissioner, or do you want to?
Maybe I just have a comment rather
Okay.
than, and then I'll defer to some of the, I know people on the floor are interested, but we probably shouldn't have sat you next to each other. We should have put Jonathan in the middle, because Jonathan's slide that really struck me was the components of a health surveillance system being eyes on the ground, the surveillance system, and then labs to do diagnostics, and then information management, and then communication outwards. And that just really struck me, is that the stranding network is this phenomenal people on the ground doing the observations. NIMS as a health program is missing the laboratory part, but USGS has a national center with microbiology, virology, and there've been some partnerships, especially over influenza. And then the information management and sharing is essentially IOS. So we could swap you around. But I think as we look forward to maybe recognizing these are our sort of skill sets, the expertise, and then how do we move those partnerships built on trust forward, I think could be, we can kind of get somewhere with, rather than trying to have folks that don't have the right ingredients to do the wrong box. So really just a comment, but
Thank you. Are there questions from the Committee of Scientific Advisors? Yes, Robert.
Yeah, thanks. Thanks for all the presentations and kind of the discussion of One Health and expanding marine mammal health to include oceanography or oceanographic data. All of that sounds fantastic. But I also see a huge number of hurdles to cross in order to get that implemented broadly. I think about, well, Francis, you just said trust, and I just really appreciate Carl's comment yesterday that progress depends upon the speed of trust, and so making marine mammal health a really kind of integrated program with people from lots of disciplines introduces trust issues, and especially kind of wildlife biologists seem to have a challenge sharing data, sharing samples, and I think oceanographers do that much better, and so to me that's one of the big hurdles, that if it's going to be veterinary health and biologists working together, that biologists really need to move in a positive direction. Michelle, I think you talked about capacity building. Maybe it was somebody else, I'm not sure, but there's a need for more people on the ground. And of course, my perspective is Alaska. And even though there's a stranding network in Alaska that, it covers really a very small area and only at certain times of the year. And so how do you develop new people and opportunities to collect more samples broadly, deal with the samples appropriately and all of that? And so kind of that introduces some questions about training and getting permits in a relatively quick period of time. How is everybody analyzing samples in a way that make them comparable? So anyway, I guess my question is the ideas are great, but and maybe it is really good for places where there's lots of people and there's lots of infrastructure and roads to get to places. But what are some ways that this could be implemented more broadly in order to make some positive progress? And I don't know who wants to answer that. So
wildlife terrestrial.
Oh boy. Okay. I guess I've been thrown on the bus. So I think there's lots of things come to mind, and I can maybe share some examples that we learned mostly through experience with whispers. because we have the same sort of concerns and issues with data sharing and trust. And I think the kind of key lessons that we learned was involve all the partners from the outset in designing these systems, that the more they feel like they've been a co-creator or a co-producer, the more likely they are to support it. You very much have to address the data privacy concerns and data sharing concerns. And so we had long conversations about, okay, what data are you willing to share? What's enough that it's useful, but not share data where you feel like you're losing control over that data. And actually, part of Whispers, we created this, it's basically a collaboration platform. So partners can link up, like Facebook, in that system, and they can share more detailed information. For example, they're working together on an outbreak. So having different layers of approval for data. I think you have to make the system useful. They have to have a benefit from contributing data, and you need to make it easy to use. And actually, that's right now what we're focusing on, is the user interface of Whispers. I think taking a step back, though, I've worked a lot of One Health projects, and myself and a colleague from the CDC and the FDA, we did a sort of review, a qualitative review of different One Health projects. We looked at avian influenza surveillance in wild birds, government response to the H1N1 pandemic several years ago, the foodborne outbreaks of E. coli linked to deer and spinach. And what we found was we have the technology to solve all these problems, but what made the project successful was very leadership skills. There was a governance structure that coordinated the work, that could then be a forum for resolving conflicts that always inevitably arise in these types of partnerships. Everybody had a shared mandate and a belief in the mission. There was equity in terms of participation. No one group dominated decision-making and so forth. Decisions were science-based and so forth. So I think emphasizing those leadership skills, training in those leadership skills, really embedding them in the networks that they're created, I think could be a factor for success. Thank you.
A slightly different perspective, not so much on the data sharing, but you mentioned sort of capacity building, and yes, marine mammals are always in remote areas, maybe not in necessarily California, but Argentina, Chile. So I think we have a living example here, but and I know in the audience we have some professors from vet schools. There is a whole cadre now of vets coming out of vet schools that are interested in marine issues, but they don't know how to get involved. And so it's sort of a two-way process. If an experienced field biologist is willing to take a young vet who's maybe trained in terrestrial wildlife but not marine, we can start to get some of those people going. And that's been a hurdle in a lot of places where there are literally young vets that just cannot get access to marine mammals, and the field biologists are sort of like, well, we don't need vets. We've been doing this for 30 years. Why would I need a vet? And then when they actually do get one, they start to think, oh, this is great. They give it a drug and it's easier. It's amazing. So it's a two-way process. And I think, again, it comes to communication, to trust. But in my experience, there's still limited contact between the vet schools that have a lot of resources. Perhaps I'm looking at Chris Croyder on the screen, but she's an example of someone who's based in a university at the One Health Institute, University of California, Davis. There are a lot of young people there who are very interested. So if they could be partnered with going out on the harvest, working with communities to collect samples in a certain way that are more useful for disease diagnostics, those are all possible.
Yes.
This isn't necessarily a direct response to your question, but it popped in my brain when Francis was talking about building capacity. So several of us were just in Alaska touring Savunga and Nome and up in Ukqiavik. And there's some really significant food availability and food security issues there. And they were talking about, their subsistence harvest and being afraid to eat what they were catching because they didn't know, whether or not it might have toxins from a harmful algal bloom or something else. And we were talking about the possibility of trying to develop a test kit so that they could use that to test the food before they ate it. Something like that would help increase the ability to gather information substantially if we were able to do something like that. And I don't know what the feasibility of something like that would be, but it would augment the availability of data if something like that was able to be developed.
Yes, Robert.
Yeah, Kim, thanks for that suggestion and talking about subsistence hunters testing samples and doing science kind of again raises the issue of permitting. And it's incredibly difficult to get a marine mammal permit if, I mean, subsistence hunters can go hunt animals and have the parts in the freezer or refrigerator. But when they start doing science, it becomes a little bit more, in my experience, is a gray area. And so I know it just takes such a long time to get the right authorizations to collect, hold, and then analyze samples, that it seems to me like that is a major hurdle, at least for dealing with these kinds of things in Alaska, and having subsistence hunters be more involved in the science.
Hey, if there are no more questions, oh, Vera.
I really like the term marine health, which is an indigenous that we already have as an indigenous viewpoint. I always hear the term that if our marine mammals are healthy, so will our communities and humans be healthy too. I really like Robert's comment about our hunters' ability to provide the results. What we're lacking in our region, Bering Straits, and I'm glad you went to Savunga, that's my home, is the capability to have the rapid testing capabilities, and also the response to the samples that we provide. We still have that lag to know if our animals are healthy to eat, because yesterday I think I mentioned that 80% of our diet comes from marine mammals. So that capability to get results in a timely manner is still something that we really need in reference to our food security.
Jason.
Okay, so this afternoon, we're going to talk about stock assessments. And one thing that is different about stock assessments than what you guys have all been talking about, at least in the National Marine Fisheries Service, is that if you look around the country, every single science center has dedicated people sustained that are working on stock assessments. But it's rare that we have a Michelle Barbieri or others around the country that are embedded and constantly and sustainably working with the programs to work on health. And so my question is, how do we institutionalize that? And Francis, you mentioned, one pathway for bringing in, young people who are keen. But a lot of times the health programs that I see with marine mammals, at least in our agency, are kind of one-off projects where you do bring in somebody like that. Maybe they do their master's or their PhD and it's gone, but that's really not going to fit with the model that you're talking about. So how do we make that transition? What are the obstacles to having that realized?
I'm going to defer to Kim, but I could have I don't work for NOAA, but so I will just say happily, I think NMFS should hire a vet PhD lead in each science center, but I don't work there.
That would be lovely if we could do that. I think it comes down to sort of the way that things are prioritized. Our funding is very limited, and so we're trying to stretch it as far as we can. And I think with the NDAA language around health map, that puts an emphasis on something that hasn't necessarily been emphasized before. And if we could get some appropriations along with that, then we could make that a high priority and be able to hire vets in every science center. But right now it's just a matter of a lack of resources, I think, that constrain the ability to do something like that. It's not a very satisfying answer. I'm sorry.
Okay. Sue and then Jonathan.
Yeah, if I could just add by example, because I did work for NOAA for 20 years. And back in 2005, we had a workshop at Woods Hole, and the focus was on passive acoustic monitoring, and that we didn't have that capability. And so myself and others, Sophie Van Paris in the Northeast, we basically worked to recommend that to the leadership here. And now I'm happy to say the science centers, all of them, all the regions, have fairly robust acoustic monitoring programs. We'll hear from Aaron later today, I think, on that. But that was key, Jason, was to get that capacity, get that capability at all the science centers. And it really was a game changer. So I just wanted to mention that example. Thanks.
So I think we have the same issue in the terrestrial systems. And I think one of the key things for me is we need to build a business case for that funding. Why is this an urgent issue? What are the contributions it will make? And what's the vision for the future? What is it going to be achieving? And I think through those business cases, we can help generate interest in funding. I think also that's where non-governmental organizations can be very helpful in terms of their ability to communicate to Congress and communicate to and engage the public and get support for these types of ideas. So I think that's one strategy that could be considered.
Right. Andy.
Yeah, I just want to maybe put a marker down for our discussion this afternoon. And Jason, thank you for bringing that up, because I think, what we've heard this morning is the clear linkages between climate and health, and in some cases, health and demography. And so the demography then feeds directly into the stock assessments. And I'm thinking, I'm thinking like, oh, why don't we have a health person in each center? I'm thinking about in Northeast with right whales, where health is a critical issue. But you can make that case pretty much in every center now. So I think when we have that discussion this afternoon about how we're going to integrate climate into stock assessments, that link through health might be a way so that we can make the case for appropriation so that we can support that kind of expertise in each center.
Great.
I also jump in and just elaborate a little bit. Thank you, Jason. I think it's also really important to recognize that having that veterinarian in those regions and associated with those programs is really, really important, but that is also just a start. So, I have the fortune of sitting here today, but back at home, there's an entire team of folks that are able to do a lot of the sampling that I talked about today. And we have a veterinary technician and a laboratory technician combined in one position who is managing a specimen archive that takes up 9 to 10 ultra cold freezers just for monk seals alone. So we do also have to recognize that there's that health expertise that we can provide as veterinarians, but we're members of a multidisciplinary team that also needs to include that larger structure and funding for those things as well.
I had one more too. Yeah. So also I will say that at the workshop, we actually had Terry Rolls and Denise Gregg had been communicating with the different science centers and kind of taking an inventory about, what are you doing related to health and just kind of starting that communication. So I think that's a good first step. I know they've continued the meetings since then because I've sat in on at least one. So, yes, definitely need more funding to do more in the future. But in the meantime, I think just coordination and communication could help too.
Absolutely. Nothing else from CSA or commissioners? I'd like to open questions for the staff on the floor. Dr. Rolls.
This wasn't planned at all. So I agree with everything that's been said, and it's been a fantastic discussion. But one of the things that I'm still trying to figure out is how do you link across these from a data management and then an active intelligence and trust building? Because we're building up from the stranding network. We have the USs. The USs don't necessarily overlap with stranding networks. And then we have the climate services group that don't overlap with any of those. Then we have the state participation. So Jonathan, I know you've dealt with this and how you start to link all of that together with the building the trust and being able to use the data to inform a lot of things like NMFS management, for marine mammals, but also a whole bunch of other things. And then I want to put a plug in for, we've talked a lot about environmental data and Sue brought up heat. We have a large heat and health initiative that Dr. Rick Spinrad is leading. As a matter of fact, at COP28 coming up in December, there's going to be at the US Pavilion a lot of discussion about heat and health, but it's really focused on human health and it's not ocean focused. And so one of the things that we're trying to do internally, as many of you know who have been NOAA employees, one of the beauties of working for NOAA is it's diverse and everything you really want and need is there. It's just focused on people, not on our ecosystems and diversity. So as we're talking about folding in the USGS and Carl's program, you need to be really directing requests to the climate services group so that we can get that kind of information to inform what we need in a timely manner, and even what they're rolling out for climate services to other nations. For marine mammals, we don't get that information directly. You have to hunt for it. So anyway, I'm just making a plug for the NOAA initiative of Climate and Ecosystem Fishery Initiative and our climate services initiative dealing with heat, air quality, water quality, and those types of things also can help inform marine mammal health. And then Jonathan, how you knit it all together, that's to you.
Well, first of all, I need to sort of respond. We're not there yet. We have grand plans, but we're definitely not at the level where we're integrating disparate data sets and doing these wonderful analyses. But I guess the thing I'd add to what I mentioned earlier about sort of networking leadership skills, I think, is patience. So Whispers went from conception in 2012 to actually finally getting funding in 2022. So it took 10 years of a labor of love, really, to keep that moving forward before we finally got the funding. So I think one of the things is it's going to take some time, right? And I recognize there's an urgency here, but you know, I think that patience, not giving up, continuing to move forward with these ideas. I think to answer your question, though, I think for me there's kind of three key components to making sure things are successful. One, you need to have the enabling environment for it to be successful, the political will. You know, the support for it. You need to have the institutional capacity for it to be successful, and you need to have the individual skills and capacity. And I think, you know, things to think about is, well, obviously if there's legislation, times I've seen federal government work well together is when they're mandated to work well together. So federal legislation saying, yes, you shall work together and do this can be very, very helpful in moving these things forwards. Absent of that, it has to be this networking, it has to be this partnership, building that trust from the ground up. The second thing is, obviously, for institutional capacity is the technological barriers. You know, as I mentioned, there's a lot of very unglamorous work that has to be done to merge these data sets. And you just need the dedicated staff, the people, the skills to actually go out and do that and start developing all the data dictionaries and data structure and the APIs to move the data in. It's not sexy. And unfortunately, people don't like to fund it, but it's absolutely essential. Although I will say, I think I was speaking, and I'm not a bioinformaticist, but I was speaking to a bioinformaticist, and they think with machine learning, artificial intelligence, we may be able to get past some of these technological barriers that used to exist, and can train algorithms to recognize the different wordings and the different ways that people name diseases to actually merge these data sets. So we're maybe on the cusp of some more advances. I think the final piece is it goes back to the question over here. You need to have the dedicated staff to do this. It's going to take a team to manage these data sets, a team to manage the data, a team to manage the partnerships. You know, like the National Wildlife Health Center in its surveillance network probably has about 30 people all working on different components, right? The field epidemiologists communicating with staff, the bioinformaticists managing the data, the diagnosticians running the diagnostic samples, the people doing the analysis and the communications. So really looking at what is that team that's necessary to make that successful, I think would be critical. Sorry, I don't think there's any magic sauce to this one.
Harry, do you have a follow-up? Question over on this side.
Hi, same as congratulations on your presentations, very interesting in detecting this relationship between climate change and health and of course stock assessment. But this is a question more to Francis about what about other indicators that could be sort of vectors of this kind of diseases. For example, all these phenomena are taking place in South America, for example, related to avian influenza. And in Mexico, with this increasing events, anomalous events like El Nino being more frequent, El La Nina being more frequent, we have this income of individuals. No, it is sometimes it's not just at a population level phenomena, but individual vectors that are being more frequent. Species from the Galapagos, for example, coming to Mexico at an individual level. We would have in recent years Galapagos lions in Mexico, some Galapagos fur seals, and maybe more important that it's related to all these die-offs in South America, southern elephant seals. Before 2020, we had not had southern elephant seals in Mexico, but from that year until now, we've had about 5 or 6 southern elephant seals. In fact, there was one adult male that stayed in the Gulf of California, between Central and Upper Gulf of California, an adult male southern elephant seal, and stayed there for a year, has stayed there for a year. And we had another southern elephant seal in the Pacific, very close to the colonies that are part of the California current system, a poor body condition adult male, subadult male. So what would be your opinion on these individual cases? And if there is some kind of advice that you could give us, in order to maybe try to sample some of these individuals, not to wait for their being stranded, because sometimes they are just resting and some have stayed, some go away. But this is something that is becoming increasing in our areas.
Thank you for that. I think that comes into this sort of umbrella of pathogens moving around with changes in distribution of the host or the vector. So, so I think, yes, they pose a very real risk that you could have a pathogen taken from where it's endemic and the animals around it have some resistance or immunity, and then it gets taken to an area that it's not. So we have 2 examples. I'm thinking at Michelle in Hawaii, where one year a northern fur seal suddenly appeared on a beach in Hawaii. So whether it came from Alaska or California, we don't know, but it was a fur seal. And another example was a northern elephant seal suddenly appearing, and it looked like a strange monk seal, and everyone realized, wait a minute, this is not a monk seal, it's a Hawaiian, I mean, it's not a Hawaiian monk seal, it was an elephant seal. So it's 2 examples of sort of extraliminal or it being. So what do we do? Well, in both those cases, I think we have pretty good disease surveillance on elephant seals and northern fur seals, because many of them strand along the California coast where the stranding network is relatively well developed. And then northern fur seals, there's been long-term sampling of animals during the harvest. So because of that baseline information on those 2 species, we already knew that leptospirosis is endemic in northern fur seals and that there's a high level of carrier status with antibodies. And then we knew for elephant seals that there's a lungworm, Otis strangulus, that can be fatal. And both those could be carried by individual animals. So we had that background, and then those individual animals were immediately handled and sampled. And then in those cases, they were actually removed and sent out. But I think that you don't necessarily want to be moving animals, back to Galapagos. But I think the disease surveillance part is sampling that individual, but also realizing that if you knew exactly what diseases were in the source population, then you would know, you would have a better handle on your risk. Is there a disease in Galapagos sea lions that you don't want to get into Mexico? And that only comes from getting some baseline data. And I suppose also communicating that, sharing that. So there's a ready way for you to know what diseases have been detected in Galapagos, which comes to the informatics and vision for, there's a map of where leptospirosis occurs in marine mammals globally. Does that help?
Chris wants to name.
Oh, Chris, sorry.
Chris, did you want to say something?
No, I just am completely in agreement. I think in many ways, the marine mammal stranding system and disease investigations that have gone along with that are really the sort of tip of the spear in us understanding diseases in general and wildlife. There's just not as much of a comparable effort and level of understanding and coordination across the network in other species. So I think, to Francis's point, that knowledge is imperative for understanding how these risks are going to be changing with climate and environmental changes that we know are imminent. So I can't emphasize and support the idea enough that we need to have a broader integrated sort of veterinary services component that is certainly in the US a huge baseline for information and support and knowledge and expertise, but extending that to other regions of the world is something that the US should really be also leading, because I think coordinating a network and collaboration gets to the other point that there are many interested colleagues, veterinarians and junior researchers in other countries that want to work on these issues. But there's not right now this fostered network and collaboration with vet schools in other regions that can support similar work. And I think the Latin America communities were largely caught off guard with the high path AI outbreak and didn't necessarily have the capabilities to respond in a way that was going to be safe and secure and had the readiness that's needed. So that's just, I think, a harbinger for things to come. So yeah, thank you for the opportunity to add a comment.
Thank you, Chris. Yeah, Sean.
Since I was recently reminded in our own program this summer when the freezer that I apparently was responsible for died, and we found hundreds of stomachs going back to the 80s of various cetacean species that are all fisheries bycatch. I just wanted to, I didn't hear it mentioned much today, but just call out the sort of the baseline opportunity that one thing this has going on all the time is fisheries and observers and bycatch opportunities as you're sampling baseline. So we have these, this sort of baseline opportunity. So just for comment or reference for thinking about this.
Yeah, I want to get a little bit more to the implementation side, sort of following up on Robert's question. The basic question is sort of detail and complexity. And so, Lori, in your presentation, you began to really line out, some very specific things we want to look for, measure, archive, surveil, right? And then we have health map, which is presumably the delivery of such surveillance information. And I think for me, that was very clarifying to see it just laid out simply. We're looking for these diseases. We're looking for this, that, and the other thing. And in the both the primordial health map things, they were very simple. And Jonathan, you put up there are those dots, and I assume you can click on those dots on the map and get some level of information. But my understanding is deciding on those levels of information and what to measure is a real hurdle for health map development. So how do we streamline, how do we push through that, oh gosh, if we're going to put this up, we've got to have everything there kind of mentality versus what's the most useful information to get out there for whichever community it is, and to assort those levels of access and information. And that goes to everybody who's been involved in developing health map as well. Just so you know.
Yeah. So first of all, I too was very impressed with the outcome of that workshop. I think those prioritized exercises, what ecosystems, what species, what diseases we should be monitoring for are critical, because as you say, we don't have the resources for everything. So I think that's a critical step in the process. I guess one thing I'd go back to is I guess we approached it is sort of like the data gets translated into information, which is what WISPERS is. It's based on an information system. We're taking that data and making it useful to somebody. The next step is actually wisdom. We actually analyze that data and have sort of some sort of analysis. The final step is decisions. You then take that information, that wisdom, and you make a decision, or what they call health intelligence. So I think maybe one suggestion would be to start from the top of that pyramid and okay, what decisions are we going to be making on the basis of this information? What are the management actions? What are the interventions? And then once you know that, you should then be able to go back and think, oh, what data do I need to make that decision? And then you can then design the system to manage the data to get you the decisions that you want. So that could be a potentially useful model to sort of hone down on what precisely is the data that you need for the decisions you're making, if that makes sense. Thank you.
One thought that occurred to me after I saw the pictures of the walruses on the beaches yesterday, and I just had this kind of sick feeling about high-path avian influenza coming up on the flyway and dead walruses anyway, and it just kind of made me feel ill. I talked to Michelle and she says, I already feel ill, thinking about monk seals. So here's an example that we're living right now of a virus that's emerging. And the only way we know about it up here in the US and for managers who have got pinnipeds they're responsible for is kind of Chinese whispers of emails. There's some stuff on ProMed. I rely upon Rafaela in Alaska to screen ProMed and send an email list to a list of people on an IWC group. So everything is very ad hoc. There's ad hoc communication. So what would you really want to know? You'd want to know, where is the closest pinniped to me that's got this? Who's working on it and the time frame. It's probably not that complicated. You don't need all the details, but just knowing whether it's in Argentina or Chile, has it got to Mexico yet? I mean, that would be. So I think we can maybe really use this current scary coming virus as a way to think, what's the minimum that we would need? What would we want to get up on IUs to let people know in California, rather than having a manager for NMFS who's about to sample seals saying, what should I be getting? They should be able to all access that information. And if we look back, the stranding network really was solidified because of a viral epidemic in 1987. And that led Joe Gerasi and his colleagues at the time to really think about what should be in the MMPA. So we kind of have another crisis, and maybe we can again turn that into an opportunity to what does that mean for Health Map?
If I may, Jonathan's answer triggered another part of my question, which I forgot, which is the question of trust. It seems like if you take that top down of what information do we really need, simplify that perspective, then the researcher who's got a zillion things they want to do with their data can say, okay, if I pull out that piece, I'm not threatening my entire research project. I'm actually contributing in a way that's not threatening. So that, I think that perspective comes from the stranding networks and the stranding partners of, oh my goodness, what are they going to do with my data? So it's also a trust thing to simplify what the requests are, and in a way simplify the vehicle that you're trying to get the information out in.
Yeah, no, absolutely. And that's precisely the approach we talk with whispers. What's the minimum amount of data people are willing to share that's useful for everybody else to see, to visualize, to have available, but not share everything they have, all the detailed data where they feel like the loss of control over that data. So yeah, I think absolutely. I just wanted to also add to Francis' point, and I think it's well made, is what we did with whispers. We took an iterative approach, right? So we had 40 years worth of mortality data. So let's make that available. It's purely for situational awareness, like she described. You can track a spread of a disease. People see it's useful. People begin to contribute. People see that it's valuable. Then they may be willing to support more funding to develop the next phase of it and the next phase of it. So I guess what they call it, minimum viable product, right, in the IT world. So start small, build what you have, and then watch it grow.
Carl, did you have?
Yeah, I didn't know that term that you just used, minimal viable product, but I've been out and about in terms of just talking about oceanography and biological, chemical, physical, all of the features. And we're a couple of decades or more behind the weather enterprise and the meteorological community, and the sampling that they have for what they do is extensive compared to the dearth of data that we have. And I'm trying to coach our marine observing and all the science experts to be less afraid to establish something that has some utility and be honest about the uncertainties and call it something with a version number. And say when you're going to try to get the next version number and say what improvements are going to be in the next version number. And that starts to help at least create some pathway and momentum, and you can be clear, bright-eyed about it. It's like, we don't have the resources to make it what we want it, but it is here and it has one question being answered, and the next one we'll try to add to it is this and then, and when it would be.
I'll say too, that, I mean, just, from the bottlenose dolphin perspective, and what has been accomplished in getting collaborators to work together and share with that, a lot of that due to Randy Wells, who started the first health assessments and has been a willing collaborator over time. But it was because we built a trust with people, and the MMHSRP program, and specifically Terry, kind of built this vision of what we can do to understand, the things that we say we want to understand under Title IV, which are the chemical, biological, and physical factors that influence health. And because we were building that data, when the Deepwater Horizon oil spill happened, we actually had some baseline information to look at, what should the health be and what is the health. And that was what helped us to determine the injury to bottlenose dolphins. And we had to extrapolate then from bottlenose dolphins to other species because we didn't have that health information. We couldn't collect the same health information. So just saying that, I mean, a lot of it is just finding good collaborators that can learn to trust each other and see the vision of what you can accomplish, pulling information together across multiple populations versus just trying to answer the specific questions for one population. Doesn't mean you can't do that. I mean, you certainly should do that, but there's just so much more power in pulling things together across populations and really doing epidemiology to try to understand what factors are that are influencing the health.
Good point. Kim?
I was going to say, I agree with what Carl was just saying, and I think it's really important in that iterative process to identify the audience. So, what the veterinarians might want and need might be very different than what the subsistence harvesters need or someone else that's using that information. So I think if you're taking that approach, it's really important to identify with that first phase what the audience is that you're targeting the information for.
Yes, and you can do masking of certain data, or some data has data, latency is different for different users. So we have some private sector companies who provide weather data, and it's not available to the public until after that company's ability to monetize that opportunity, but then it goes into the operational forecast, for instance, to make the forecast better. So it's a win-win in that situation. So you could create a vet-only access point where some specific data may not be available to others because you have a reason for that. You don't want to create a bunch of walls like that if you can help. It's better to have open data as the philosophy, I think. And we have in the ATN, the Animal Telemetry Network, we're having to change culture to get people to realize that the world doesn't fall apart when your data is available faster. It usually actually leads to greater benefits.
Absolutely. Brady, you had a question.
Hey, this is Mike on. Okay, great. This is a question from online that Chris actually responded to. So the question or the comment online is, in addition to monitoring animal movement and population demographics, increasing our understanding of changes in congregation patterns, both intraspecific and interspecific, will be important to evaluate future risks of certain diseases to marine mammals as the environment changes, morbillivirus virus, influenza, brucella. So Chris, you agreed. Do you have a further comment on that, or does any of the other panelists who didn't get to see that online want to say anything about that?
I would just add, I think it's an excellent point and a huge task, as has been stated for many of the things that are urgently needed, but we can expect to have more emerging infectious diseases such as high-path avian influenza that will affect and potentially have really dire consequences for populations, especially smaller at-risk conservation risk populations. So I think this type of work and modeling and collaboration across a lot of different folks who look at their own species of interest and trying to understand how those global patterns are supposed to not only overlap currently, but expecting what that will look like in the future. So I think there's a major role for not just collaboration across many species, but with other disciplines that can help us with the large data problems. It was already alluded to that I think there's a lot of innovation, obviously, in the AI, artificial intelligence space, and machine learning. And I think that is an opportunity for us to try to get a grasp for some of these huge data problems, like I think this would entail. Thank you.
If I could just say something with regards to the telemetry side of things. We are still so far behind the curve in terms of knowing the movement patterns of so many of the marine mammals that are out there. We know distributions, we know the ranges of species, but the individual movements within those ranges, the potential vectors, the degree with which individuals move from one site that could have a disease to another site is really just at its infancy for so many of these animals, especially from my experience, small cetaceans. But it is an area that I think can provide us with a great deal of information, specific information for how some of these vectors might work. More questions? Yes, Sean.
If the commission wanted to make a recommendation on that to the agency, that would help me a lot. And Carl.
Carl.
Looks like something I said yesterday, and I might have appeared smart, but I wanted to give attribution to Mark Osler, who I think is the one who said it first, that made me say progress moves at the speed of trust. Another thing that he has said, working in the coastal resilience realm across agencies to try to build services that are helping people every day at the coast, is that partnerships mean he said partnerships move forward at the speed of trust is another way I've heard him say it. And then with partnerships, he taught me this, and I love this, is that when you're partnering with someone, it's really about committing to what you're not going to do. And so that's where you have multidisciplinary teams getting together and you figure out, okay, you've got this, I've got that. And that's a little bit to Terry's question and comments earlier. I was thinking about that comment, and I wanted to give Mark Osler credit for what I said yesterday and give you another angle on that on the partnerships front. And on back to the questions on the screen, we've got a good partnership going with sanctuaries and IEAs where we've got a webinization of biological data is what we've been calling it for years. And so that's a potential platform to leverage, and that Kim's team's working with our team on to potentially use for some of the products and visualizations that you might use in the health map context.
We're at the 2-minute warning. Do we have more questions? Yes. Kim. Hope we're recording that.
I just have a question for you all. If a state is trying to build this type of whispers project, do you all have a framework that we can follow that will be beneficial with building that trust? When you have biologists that are doing one thing at one university, a veterinary school is trying to do one thing, coastal people that are trying to do one thing, so that we don't have to reinvent the wheel, we can use what you've learned and try to start implementing it on the ground.
So if you're talking about the code for the system, that was all publicly available. So anybody who wants to take that code and build their own system can do so. So if you're interested, I maybe we can connect afterwards. But I think your question was more, is there a handbook for building trust? Is that? Okay. You know, I'm not aware. I don't know what anyone's aware. There's books out there, right? And there's materials, but there's not unfortunately, it's not a guide. I don't know anybody else. Sometimes you learn the hard way, you know. Go ahead,
Peter.
I'd interpret the question, having looked at whispers, you have state agencies participating in whispers, agencies that are dealing with wildlife health, different vectors of disease and stuff. And so I'd say the first place for someone in a state to go would be to their state wildlife agencies and see how they're playing in the national wildlife, and to USGS people in their state, see how they're playing in that kind of wildlife health center network stuff. That would be my sort of direct interpretation of what to do next.
Yeah, and I think a lot of states are building up their own like One Health networks, One Health platforms, and that may be also a great venue for a lot of these discussions too. So there's an ability to tap into or connect with those One Health networks. That would be great start too. Thanks for the clarification.
And I would like to thank the panel very much for responding to the excellent questions from the audience and the commissioners and the CSA. And very informative. And I turn the mic over to our illustrious leader.
No, I'm just going to be very strict and say at 1:15, we're back and starting because we do have a limitation. We do have to be out of here for sure by 5. Every panel is going like perfectly, so I'm not really worried about it, but just think about that as you start the afternoon. And I'm also curious if anyone can get us sort of a 1-page thing to put up about this avian influenza outbreak in South America. As you guys have all referred to it, you all like know what